Muchas gracias. Me están diciendo algunos alumnos que están aquí al tanto de la transmisión que parece que ya se reinició. ¿Es así? Ok, muy bien. Bueno, una disculpa, parece que hubo un corte en la transmisión, pero parece que ya regresamos. No sé bien en qué parte nos habíamos quedado. Había hablado de la epidemia. Aquí, ok. Había hablado de la nueva variante que fue reportada por el doctor Robert Will, ¿verdad? Que fue por mucho tiempo el encargado del sistema de vigilancia epidemiológica de Edimburgo, del Reino Unido y de toda Europa. Sí, él realizó muchos esfuerzos para precisamente llevar a cabo esta, esta, estos estudios en toda Europa. No sé si ya se volvió a cortar la transmisión. Vamos al mismo, ok, perdón. A ver un momento. ¿Cambió? Creo que cambió, ¿verdad? Sí, ya me mandaron un mensaje que se cortó la transmisión. Entonces, este nuevo enlace, si el problema es pasárselo a todos los demás que están en las transmisiones. Y ahí voy. Es que como cambió el URL, ¿cambió o sigue siendo el mismo? No, ya lo reenvié a varios grupos que me han comentado que van a estar conectados. Una disculpa otra vez por la situación, pero parece que pudimos retomar. Como comentaba, estamos en un nuevo link, ¿no? Pero bueno, había comentado que el doctor Bob Will es quien realiza estos estudios para identificar una nueva variante en los humanos, que es producto del consumo de la carne contaminada. Un artículo que publican en 1996 y había pasado a comentar sobre el proceso de tipo iatrogénico, que es producto de una transmisibilidad por intervenciones médicas. Aquí fue una serie de trabajos por varios autores, por eso los puse como en un clúster por separado, en el que se reporta tanto transmisibilidad por trasplantes de córnea, electrodos intracerebrales, hormona de crecimiento, injertos dura madre y transfusiones sanguíneas, ¿no? Son los mecanismos de transmisibilidad que se han encontrado hasta este momento entre humanos, ¿verdad? Además de, obviamente, las variantes hereditarias. En algunos hallazgos técnicos que se mencionan en esta diapositiva, ya hablamos de algunas variantes más que nada relacionadas con hallazgos relacionados con la enfermedad, la transmisibilidad del scraping, el hallazgo de Prusiner, pero hay otros hallazgos que han ayudado a entender el mecanismo de transmisibilidad de las enfermedades por priones. ¿No? Se empezaron a encontrar cepas o variantes de estas proteínas. Se empezó a encontrar que la proteína celular aparentemente tiene como función controlar la degradación y mantenimiento de la mielina, que es una cubierta que protege las proyecciones de las neuronas para facilitar la transmisión de sus impulsos entre una y otra, pero también otros hallazgos técnicos como el PMCA, Protein Misfolded Secret Amplification, que es un estudio o una técnica que permite hacer uso del prion normal, ponerlo en contacto con priones normales, los celulares, y observar cómo eventualmente generan un cambio. Este hallazgo es prácticamente la evidencia eh, técnica experimental que puede pues, limitar la suspicacia de las personas que sospechan que el prion puede inducir cambios y generar su replicabilidad. Y esto llevó, no aparece en esta imagen, a que entre el 2008 y el 2010 empezaran a generar una nueva estrategia diagnóstica que se conoce como RT-Quick. Es una especie de ensayo muy parecido a este. Se utiliza tejido de una persona que está, en la que se sospecha una enfermedad por priones, se inocula con una siembra o semilla, como se le conoce, de priones normales, se deja un tiempo bajo condiciones específicas para que el prion empiece a replicarse y se empieza a tomar una muestra a otros tubos y este proceso se repite una, dos, tres, cuatro, cinco veces, en el cual se empieza a ver que gradualmente la cantidad de priones anormales es mayor, ¿no? Y esta es una variante de este PMSA que se llama RT-Quick, que ya se ha sistematizado y que permite realizar un diagnóstico mucho más específico de las enfermedades por priones al que se realizaba anteriormente, ¿no? Creo que con eso tenemos una visión global de cuáles han sido estos pasos cruciales. Puse cumbres muy grandes, ¿no? En las enfermedades por priones, desde la primera descripción de una enfermedad en las ovejas, el scrapie, al que tenemos, a los que se realizaron por los investigadores, Kreuzfeld, Jacob, Gettman, Stoller, Schanker, Gagdusek con el Kuru, Pierluigi Gambretti con el insomnio familiar fatal junto con Lugaresi, Stanley Prusiner para identificar el prion, doctor Bob Will para la nueva variante, y el conjunto de investigadores con las variantes iatrogénicas, ¿no? Y está listado tanto cuanto técnicos que han permitido avanzar en el conocimiento de la proteína, en el diagnóstico y eventualmente para poder generar nuevas moléculas que pudieran servir como tratamiento, que también será el enfoque de alguna de las presentaciones posteriormente, ¿no? Y yo con eso termino agradeciéndoles su paciencia para estos cortes, ¿no? Y también el tiempo que le dedican para conocer un poquito más sobre cómo se ha ido avanzando en el conocimiento de estas enfermedades, que si bien desde hace 300 años tenemos el primer registro clínico en ovejas, y hace un poquito más de un siglo en humanos, los hallazgos fueron monumentales a partir de la década de los 50 del siglo pasado a nuestras fechas. En los últimos 60, 70 años ha habido mucha sistematización en el estudio de las enfermedades, se ha podido conglomerar en cuadros clínicos, en evidencias que permitan ahora favorecer el diagnóstico y eventualmente al tratamiento. Muchas gracias y los dejo con la siguiente charla. Muy bien, muchas gracias. Hablaremos ahora o presentaré al profesor Richard Knight. El profesor Richard Knight Tiene la distinción de profesor, que en Europa es la máxima distinción académica que se asigna a un académico o a un investigador. Em, esta, esta distinción la obtiene precisamente por haber colaborado con el sistema de vigilancia de enfermedades por priones en Edimburgo. Es actualmente uno de
Well,
los coordinadores del sistema de vigilancia de
Priones,
enfermedades por priones en Edimburgo. Fue un colaborador estrecho con el profesor Robert Will, quienes ayudaron a identificar
diseases
precisamente la
affect
epidemia
animals.
de las vacas locas, la nueva variante de enfermedades por priones. Es miembro también de la Fundación de Enfermedades por Priones en el Reino Unido. Es, eh, es, es también codirector de la Alianza Internacional de Enfermedades por Priones junto con Dina Simpson y con Susan Solvins, y ha ayudado al establecimiento y guía de múltiples centros de vigilancia epidemiológica de enfermedades por priones a nivel mundial. Ha sido también asesor de la Cruz Roja Internacional en enfermedades por priones de la Organización Mundial de la Salud. Em, a su vez de que el doctor, como neurólogo, egresado también del Reino Unido, en las universidades de Oxford, como médico y como neurólogo, con honores en ambos grados académicos, se encuentra ahora ya casi en las puertas de su jubilación, lo que nos daría mucha tristeza, pero que ha llevado una trayectoria muy importante en las enfermedades por priones desde el establecimiento del primer sistema de vigilancia en Reino Unido, posteriormente el europeo y eventualmente a nivel mundial. Sin más, le dejamos la voz al profesor Richard Knight para escuchar su presentación sobre el concepto del prión.
Well, hello. My name is Richard Knight. I'm a neurologist in Edinburgh, in Scotland, in the United Kingdom. Victor asked me to give an introduction to prion disease, which I will do. I'm sorry that I can't do this in person, but I'm in Australia at the time of your meeting. So, prion disease, what does it mean? Well, there are a history of names here. Originally, this disease was called Creutzfeldt-Jakob disease after two clinicians, pathologists who described it, Creutzfeldt and Jakob. It was then given the name of spongiform encephalopathy, which basically means spongy change in a disease of the brain. It was found to be transmissible under certain special circumstances, initially to the chimpanzee in a laboratory, and was called transmissible spongiform encephalopathy, although these days we prefer the molecular-based name of prion disease, which I will endeavour to explain. Prion diseases affect animals as well as humans. I'm not going to talk about all these animal diseases, but they have played an important part in our understanding of prion disease. So, I'm going to give some background biology. I'm going to talk about prion disease in terms of its mechanism and causes, and I'm going to touch at the end on diagnosis and the possibility of treatment. So some background biology, and I'm going to apologise to those of you who already know this. The body is made of organs like the heart and the brain. The organs are made of cells, which are little biological units with a membrane containing fluid, chemicals, and various other things, and in the centre a small nucleus. But everything is made of chemical structures, and one major chemical structure is a protein. Now, proteins are made up of a chain of small chemical units called amino acids. They're put together to form long chains. When you look at the cell, this blue thing here is the nucleus in the cell, and if you open it up, you find these wriggly things called chromosomes, and chromosomes contain genes, and genes are the codes for making proteins. Now a code, it's a string of code, the gene, and each unit in the code, called a codon, stands for one amino acid molecule. So when the code is translated, you get a string of amino acids. Now the genetic code instructs the production of this basic protein change, but after the chain is formed, there's a complex folding of the basic chain to make a complicated three-dimensional final structure. And this three-dimensional shape governs its interaction with other chemical structures and therefore its functions. In the bottom left-hand corner here you see a hemoglobin molecule, which is a protein, and you can see its complex structure, and it's designed for carrying oxygen around the blood. Well, a key aspect of life is that chemical structures interact with each other. Their shape affects how they fit together and therefore how they interact. So you can see in the bottom left here that there are two structures which can interact with each other. And on the right, there's a structure which won't interact with the main structure on the left. So the structure of proteins does have a big effect. This interaction that you see here could cause disease because of the way the proteins interact with each other, or maybe it could even prevent disease. So I'm now going to go on to prion disease proper, the mechanism and the causes. The fundamental molecular event is a protein structural change in a protein called the prion protein, and that's why we use the term prion diseases. All of us have a normal gene, PRNP, which is responsible for producing normal prion protein. So I'm producing prion protein now, as we all are. But in disease, this prion protein undergoes a conversion to an abnormal form. It's the same chemical, the same string of amino acids, but it forms a different three-dimensional shape. Now in prion diseases, it's a slightly weird event, but the abnormal protein, once it appears, interacts with the normal prion protein to turn it into more abnormal prion protein. And so once this process starts, it tends to amplify and produce more abnormal protein. So the basic disease process, you have a normal gene producing a normal protein, PRPC. In disease, it converts to an abnormal form, PRPSC. And as I've mentioned, the abnormal form then amplifies itself by converting more normal protein into more abnormal protein. Because the abnormal protein has a different shape, it's dealt with differently. It tends to be insoluble, tends to aggregate and gets deposited in tissues. And you can see here this brown staining is deposition of abnormal prion protein in a brain from someone who had a form of prion disease. Now, in some way that we don't fully understand, all of this basic molecular problem leads to neurons, brain cells, malfunctioning and dying. And it's the malfunction of these brain cells which causes the symptoms of the disease. And as a result of loss of these cells, you get holes in the brain, this spongiform change. Well, what sets this process off? There are three sorts of causes: genetic, acquired and idiopathic. Genetic first. Well, clearly, if there's a string of code in a gene, there could be changes in the string of code, mistakes or alterations in the code. And there are two important types of change. One is a pathogenic mutation. It's a change in the code which is important because it leads to a resultant dysfunctional protein. And as a result of that, disease is caused. But there's also polymorphisms, which are changes in the code, and therefore you get changes in the protein, but the protein still folds pretty normally, and the resultant protein works well enough, and it does not cause disease. It's just a sort of variation. Well, if we deal with pathogenic mutations first, Clearly you have an abnormal gene, as I've indicated with the red star. You therefore get an abnormal protein, and in some way this abnormal protein is more likely to convert to an abnormal form and therefore cause disease. In the past, the genetic forms of prion disease were given names like genetic CJD, Gerstmann-Streusler-Schenker syndrome, fatal familial insomnia, and so on. But the modern classification tends to be based on the mutation, and the mutations are given names like E200K, P102L. These are very common mutations that you see in genetic prion disease. So if we just turn to this modern mutation, the codons are just numbered from left to right, and each amino acid has a letter symbol. And so what you have is the amino acid which is affected by the mutation is indicated by the two flanking letters, and the number in between is the position in the code where the change occurs. So E200K is a change at position 200, the 200th item of code, and it's related to glutamic acid and lysine, which have the letters E and K. Now, these genetic diseases are inherited. So if you take two people, the white square is the normal gene person, and the other coloured square is the abnormal gene person. If they have children, each of their offspring has a 50-50 chance of developing the disease because they inherit the mutation. However, it's very important to understand that each offspring, every time they have an offspring, there is a 50-50 chance. So although I've shown you here 4 children, 2 of whom have the mutation and 2 of whom don't, it could be by chance that all 4 don't, or could be by chance that all 4 do, or even 3 of them do. Each time it's a toss of the coin. Polymorphisms, they do not cause disease, and I just really want to give you a kind of ordinary example of what I mean. So supposing the correct code for your holiday is the plane leaves at 2100. A polymorphism might be your plane leaves at 9 p.m. That's exactly the same information. It doesn't lead to an error in itself. You know when the plane is leaving, but it's put in a different way. A pathogenic mutation might be your plane leaves at 1900, when in fact it leaves at 2100. So polymorphisms are not directly problem-causing, but sometimes, incidentally, they make problems more likely. So, for example, if someone says your plane leaves at 9 and you don't understand the difference between AM and PM, you might well make a mistake and therefore miss your plane. And that mistake would be avoided by using the 2100 terminology, provided you're familiar with the 24-hour clock. The purpose of all of this is there is an important polymorphism in the prion protein gene at position 129 relating to methionine and valine, 2 amino acids, which have designated MV. And you can see here that about half the UK population are of the MV type, quite a big proportion MM, and a small proportion VV. But all of these people are just normal, they just have a different variation of the prion protein gene. However, I did say that sometimes, incidentally, they make problems more likely, and we will turn to this a bit later. Acquired disease. Well, in some ways, acquired disease is the easiest to understand because basically abnormal protein from an external source enters the person or animal's body and then causes the normal prion protein to convert to abnormal form. If you're exposed to something that infects you, there is a gap between exposure and the symptoms appearing of the disease, and this is the incubation period. For things like common colds, it's measured in days. But in prion disease, it can be very long, years or even decades, and that complicates matters somewhat. There are 3 forms of acquired prion disease: Kuru, a disease of certain people in Papua New Guinea, variant CJD relating to BSE in cattle, and iatrogenic CJD, which is accidental medical or surgical transmission. I'm not going to talk about Kuru, although it's historically a very interesting disease. It is now either extinct or virtually extinct and affected particular people in the eastern highlands of Papua New Guinea. Variant CJD was related to BSE in cattle entering the human food chain and infecting humans through food, mostly in the UK. It appeared about 1981 in the UK, formally recognised in 1986, and prior to 1989, when various protective measures were put in place, nearly 500,000 infected cattle entered the human food chain in the UK. It did spread to humans in the form of variant CJD, but fortunately it did not affect very large numbers of people. 178 people affected in the United Kingdom. Last case had onset of symptoms in 2014. France was number 2 in terms of numbers, and most other countries have had very few cases or none. There are no known living cases of variant CJD in the world at present. Iatrogenic CJD relates to accidental medical or surgical transmission from one person to another. Dura mater is a fibrous membrane that was obtained from people who died and used in surgery and has accidentally transmitted CJD from one person to another. Pituitary gland hormones, hormones taken from the pituitary glands of people who died and then used in medical treatments, have transmitted CJD. On a handful of occasions, neurosurgical procedures have done it, and at least on one occasion, corneal transplant. Iatrogenic cases related to cadaveric-derived human growth hormone human growth hormone. Certainly these have been reported. Synthetic hormone has been used since 1985, so the risk is no longer there, but the incubation period for CJD can be long, and there have been 6 deaths reported in the UK between 2015 and 2023, reflecting incubation periods of even up to and over 40 years. Idiopathic basically means of unknown cause. And idiopathic disease simply means that we don't fully understand why this occurs. It's also called sporadic disease because it occurs sporadically in all populations of the world where it's been studied. It appears to be a spontaneous error that just occurs in the person whereby abnormal protein arises and causes disease. It's the commonest form of human prion disease, accounting for over 80% of cases. Most countries where it's been studied, you see 1 to 2 deaths per million of the population per year, but the lifetime risk for any individual developing the disease is about 1 in 5,000. It appears to be spontaneous, but we don't fully understand why it happens. There are variations in the clinical and pathological picture, so people with sporadic disease can look a bit different from each other on occasions. One of the reasons for this, of course, is the polymorphism, and I mentioned that sometimes incidentally makes problems more likely, and your polymorphism can affect the probability of your developing the disease and the clinical picture if you do develop it. And in acquired forms, not the sporadic forms, it can affect the length of the incubation period. Sporadic CJD is a bit variable, as I've mentioned. There are differences in the clinical and pathological pictures and differences in diagnostic test results. And on the basis of this, it's been found to correlate with, first of all, this 129 polymorphism, and also with certain variations in the prion protein, which has been designated as type 1 and type 2. So sporadic CJD has been divided into subtypes, and you hear people talk about MM1 and 2, MV1 and 2 and VV1 and 2 types of sporadic CJD. But effectively, it's all one basic disease, and these are just variations in the way it appears based on variations in the underlying molecular changes. I wanted to talk a bit about diagnosis. And first of all, I would like you to understand that clinicians are trying to find their way through a diagnostic maze. The initial symptoms of prion disease are non-specific. Someone may present with certain symptoms, and there may be 4 possibilities, including CJD. And then as you go through, you narrow things down, and you finally get to a final diagnosis. From the patient or family view, this may look rather obvious. Well, the presentation was typical of CJD, and you can follow the red lines through the maze. However, it's important to understand that this is not the view as you're going through it in real time. As you're going through it, it's not obvious. The presentation is not only typical for CJD, but also typical for other illnesses, and there are many possibilities that you have to consider. And of course, very importantly, some of these other possibilities are potentially treatable, and therefore you don't want to miss them. Well, the diagnostic process in outline is you exclude these other possible diagnoses, you consider CJD, and then you think about tests which confirm or support your diagnosis of CJD. All of this can take time, which is frustrating for families, and also particularly frustrating if you're dealing with a rapidly progressive disease that sporadic CJD, for example, often is. There are 2 definitive tests you can't argue with: finding abnormal prion protein in the brain. Now, this requires brain tissue, and therefore it's done either at autopsy after death or in life through biopsy of the brain, and you might understand that biopsy of the brain is undertaken only rarely. Finding a genetic mutation in someone who's clinically neurologically ill is certainly a very definitive test, but it relates to genetic forms of the disease only. There have been 3 classical tests which are very useful: the EEG, the brain MRI, and the spinal fluid 1433. They're extremely helpful, and in the right kind of clinical context, they give very reliable diagnostic results. But the abnormalities seen in these tests are not 100% specific, meaning they can be seen in other diseases. Now, of course, if you could find abnormal prion protein, that would be a definitive test, because that is what the disease is all about. But detection methods require a certain level of abnormal prion protein. In the brain, these levels are high enough for our detection methods to work. In other tissues, which might be more accessible, they're generally too low. However, you may recall that I said the normal disease process is one in which abnormal prion protein converts normal prion protein into more abnormal form. And what people have done in laboratory tests is they've hijacked this disease process in what is called amplification techniques. So you have a sample with a small amount of abnormal prion protein in it, shown by the red square, too low to detect. But you add normal prion protein, you do various clever things, and the abnormal protein converts the normal protein into more abnormal protein. And as a result, you've now got a level that you can detect. There are 2 basic amplification techniques. I'm not going to go into detail. I'm mentioning them because you may hear of them: PMCA and RT-QuIC. There's one amplification test in routine clinical use. There are others in development, but one in routine use now. It's the spinal fluid RT-QuIC test. It's highly sensitive and highly specific for sporadic CJD. In the United Kingdom, 94% of our sporadic cases have an abnormal RT-QuIC test. And so far in the UK, all of the people who've had an abnormal RT-QuIC test have indeed had sporadic CJD and not another illness. I just want to say that detection of abnormal prion protein in tissues outside the brain using these amplification techniques does not mean that these tissues are an infection risk in ordinary life. Ordinary, even intimate contact with somebody with CJD is not a risk. People do not catch it from other people in this way. I wanted to finish on the possibility of treatment. There is no established treatment at present, but I think we can say there is now the possibility of it. A rational approach to treatment follows the understanding of the disease process. If you can reduce or get rid of PRPC, then that will, of course, stop the process or slow it down, because there is no prion protein there to convert to the abnormal form. If you could prevent the conversion process, this would also help. If you could remove the abnormal prion protein, this could help. If we understood how proteins, all this protein changes, cause neurons to malfunction and die, we could perhaps interfere with that neurotoxic process. All of these things have been considered, and all of these things are the subject of laboratory developments looking for potential treatments. And I think it's fair to say that in the foreseeable future, we will probably see some of these treatments coming through to human trials. Just one comment. The earlier the treatment, the better the outcome. That's usually a feature of most diseases, but early treatment requires early diagnosis. Symptoms may well begin some time after the disease process has actually started, and that's particularly true perhaps in sporadic CJD, which is so rapidly progressive. Is it possible to diagnose early enough? Well, we obviously have to explore that difficulty, but the more we go into treatment, the more we need early, rapid diagnosis. So I'm going to finish there, and I've been involved with these diseases over many years. Day by day, as you're in your ordinary work, nothing seems to change, but when you look back, everything is different. Our understanding of these diseases is much greater. The diagnosis is now much better, and we do have the real promise of treatment, even if we still currently have no effective proven treatments. So I'll finish there and hope that this has been of some help to you, and thank you for listening to me, and thank you again to Victor for inviting me to give this talk, and once again I'm sorry it has to be done remotely.
Bueno, muchísimas gracias al doctor, al profesor Richard Knight por su plática, que me parece que fue muy enfocada a los conceptos de lo que es el prión, cómo es que esto puede generar algunas enfermedades, ¿no? Y cómo incluso ha ayudado a generar nuevas expectativas diagnósticas y de tratamiento. Agradecemos bastante al profesor Richard Knight por esta ponencia. Cualquier duda que ustedes tengan en función de las prestaciones que hemos tenido hasta el momento, por favor, mándalas por escrito. Está ahí la posibilidad de hacerlo a través de la transmisión en el YouTube, para recabarlas, pasárselas a los ponentes y que nos puedan eventualmente dar sus respuestas. Sin más, pasaríamos entonces a escuchar la siguiente presentación, ¿verdad? Que sería del doctor Michael Gershwin. Si es que no me estoy equivocando yo en el orden del día. Sigue el doctor Michael Gershwin en su presentación de enfermedades por priones, y no sé si ya esté lista la, el, la, el banner, la presentación del doctor Gershwin. Ok, bueno, empezaré presentando al doctor Michael Gershwin. El doctor Michael, muchísimas gracias. El doctor Michael Gershwin, perdón, es profesor y neumólogo del Memory and Aging Center de la Universidad de California en San Francisco. El doctor es egresado de la UCLA, de la UCLA, Universidad de California en Los Ángeles, y obtuvo su especialidad en neurología en la Escuela de Medicina del Instituto Johns Hopkins. Ha sido editor invitado de la Academia Americana de Neurología en el tema de demencias. El principal enfoque del doctor Gershwin en sus proyectos de investigación será la valoración, manejo y tratamiento de las demencias rápidamente progresivas. Ha apoyado también a través de la clínica de demencias de la Universidad de California en San Francisco los estudios clínicos y básicos en cuanto a las enfermedades por priones, desde el diagnóstico y tratamiento hasta la valoración clínica, junto con el doctor Stanley Prusiner, que ya vimos que es el premio Nobel por estas enfermedades. Y ha apoyado también a varias fundaciones a nivel internacional, es parte del Consejo de Asesores de la Alianza Internacional para el Creutzfeldt-Jakob, el famoso CJD-ISA, el grupo de apoyo, del cual también nos hablará en un momento más, y lo tuvimos también como ponente en nuestro Centro Universitario de Los Altos hace algunos años, en donde también nos habló del tema de enfermedades por priones. Es un ponente buscado de manera habitual en estos temas a nivel internacional y nos da mucho gusto tenerlo el día de hoy con nosotros para que nos hable de este tema de enfermedades por priones. Sin más, le dejo la voz al doctor Michael Gershwin. Creo que no ha empezado, ¿verdad? Ah, ya está empezando.
I'd like to give a little bit of an outline of what I will be talking about today. I'll be talking about some of the clinical aspects of human prion disease, and I hope to be building upon the talk that my colleague Dr. Richard Knight gave prior to my talk. And I'll talk about the three main forms of human prion disease: the spontaneous or sporadic form, Creutzfeldt-Jakob disease, or SCJD, which usually presents as a classic as well as in a non-classic form. The classic form being usually a rapidly progressive dementia or cognitive impairment with problems with gait incoordination or ataxia. And then the non-classic forms, which usually present slower and can have many different presentations, usually that are more cognitive in presentation, as opposed to motor symptoms. Also talk a little bit about the use of MRI for diagnosis and how our criteria at my institution, the University of California, San Francisco, differ from those used in most other locations and in most other countries' criteria. Next, I'll talk about acquired prion diseases, very briefly about Kuru, iatrogenic, and variant CJD. Kuru being the acquired prion disease that occurred many years ago, more than 50 years ago, in the Foray ethnic group in Papua New Guinea. Iatrogenic CJD are those caused accidentally by medical or surgical procedures. And variant CJD is essentially due to the prions that originated from bovine spongiform encephalopathy, or more commonly known as mad cow disease, in which people were exposed to those abnormal prions and developed a new form of prion disease called variant CJD. And then I'll finish up by talking about genetic prion diseases. And I'll talk about briefly the historical classification and how that originated when we did not know that there was a gene that could cause this disease, but it was known to run in families. And it was thought that maybe it was passed down through an infection from person to person in the family and due to exposure, but it wasn't known that it was a genetic disease. And then once we knew it was a genetic disease that was causing the type of prion disease to run in families, it changed our nomenclature somewhat. And then I'll talk about how we're studying prion diseases, particularly genetic prion diseases, at our center at UCSF. So human prion diseases are generally divided into three main types depending upon how they are acquired. About 85 to 90% are spontaneous or sporadic, including Creutzfeldt-Jakob disease or CJD or JCD. Just as an aside, it turns out that Hans Creutzfeldt described a patient in her 20s in around 1920. And Alphonse Jacob, who was a junior professor to Dr. Creutzfeldt, later described 4 cases which he mistakenly thought was similar to Creutzfeldt's case. It turns out that Creutzfeldt's case did not have what we now consider prion disease, but at least 2 of 5 of Alphonse Jacob's cases did have what we now call prion disease. So for many years, the disease was called Jacob's disease, as well as Jakob-Creutzfeldt, but for reasons I won't get into, the name Creutzfeldt-Jakob has unfortunately stuck with this disease. The genetic prion diseases are much less common. They're about 10 to 15% of prion diseases, and they're divided basically historically into 3 types: familial CJD or JCD, Gerstmann-Strauss-Scheinker disease, and fatal familial insomnia. And later today, I'll really only talk about familial JCD as well as GSS. And in particular, for familial CJD, I'll talk about the one form called the E200K mutation that's most common in Mexico. Once we discovered that the familial forms of prion disease were actually genetic in origin, the nomenclature changed, and that allowed us to identify new forms of genetic prion disease that didn't fit into the previous clinical and pathological categories, the 3 above, the FCJD, the GSS, or the FFI. These include octopeptide repeat insertions, or OPRI mutations, and these tend to be divided and can present as familial CJD clinically and pathologically, or they can present as GSS. And then there are at least 3 mutations that are stop codons, in which a full protein is not made. A truncated form of the prion protein is made, and these present very differently than any of the prion diseases. And in fact, they look like a very slow progressive neurodegenerative disease. And then the acquired forms I'll talk about as well. Even though these are the most notorious forms, they actually are by far the least common, only accounting for less than 1% of all human prion disease. And I'll talk briefly about the curu, the iatrogenic, and the variant CJD, and how they differ from the sporadic. So human prion disease So I tend to divide prion diseases, particularly the genetic form, but here I'm talking about the sporadic form, into those that are classic or fast progressing, and then later I'll talk about the slower progressing. Fast progressing, usually less than a year survival. So an example would be a 61-year-old gentleman who had 3 months of problems with becoming confused. He was forgetful. He had problems with his vision, and he had problems finding the right words. In the first month of his illness, he became disoriented around his house. He couldn't figure out how to navigate around his own home. He had trouble reading. He had trouble reading words on his computer. When he was speaking, he couldn't find the right word. He looked at his hands and his feet, and he told his wife that they seemed very enlarged. And and no one else noticed any other problems, only him. When he looked at people's heads, sometimes they looked enlarged to him. He had to stop driving. By month 2, he became echolalic, or repeating what other people were saying. He had decreased speech output. He was no longer able to ride a bike like he'd been doing daily for many years. He couldn't use utensils when eating, and he was no longer able to help with the household chores. By month 3, he could no longer be left alone and became easily angered and frustrated. And unfortunately, he died by about 5 months after onset. And this was his MRI when we saw him at 3 months. And on the top these are both diffusion weighted sequences. And on the top, what we're showing is a diffusion weighted MRI. And on this MRI, the areas that are bright white, which hopefully you can see, are being shown. And those are on the surface of the brain, or what we call on the cortex, what we call cortical ribboning. And it might be hard to see, but on the bottom of the screen, we show a similar sequence in which the areas that are bright on top that show reduced diffusion of water molecules are very dark black. And if you see the bright white on top and the black on the bottom, that tells you that there is truly restricted diffusion of water molecules, and that what you're seeing on the top row is not artifactual but real. And this is classic for prion disease. We see in the cortex this what we call the ribbon or cortical ribboning, and then deep in the nuclei on the second image on both rows, that on the left is right and right is left here, but where there's an arrow pointing to near the center of the brain, that part of the brain called the caudate head is abnormally bright above and it's dark below. And these are classic findings for CJD. An atypical case is a patient who had a very slow progression of a disease that some people would call primary progressive aphasia. His aphasia is a disorder of language, and he had a disorder of language function. He's a 66-year-old right-handed gentleman with 2 years of progressive language problems. He had trouble finding words, he had inability to name items, he had difficulty understanding when he was listening to long sentences or to listening to someone on the telephone. He was still very fluent. He could speak at a rapid rate, but he had trouble finding words. At the time we saw him, he was still mountain biking daily, kayaking, and driving a truck daily for work, despite having 2 years of symptoms. about 10 years ago. So depending upon where the misfolded proteins are accumulating in the brain, you can get very different symptoms. And here I just show that depending if they're in the occipital or the back part of the brain, you can get visual symptoms. If they're in the area in the front part of the brain, let's say below the pink and blue areas, that's a language area, you can get language problems. If they're in the blue area, you can get memory problems. If they're in the pink area, you can get personality changes. So where the abnormal prion proteins are misfolding, that really dictates where the symptoms are. About 10 years ago. So depending upon where the misfolded proteins are accumulating in the brain, you can get very different symptoms. And here I just show that depending if they're in the occipital or the back part of the brain, you can get visual symptoms. If they're in the area in the front part of the brain, let's say below the pink and blue areas, that's a language area, you can get language problems. If they're in the blue area, you can get memory problems. If they're in the pink area, you can get personality changes. So where the abnormal prion proteins are misfolding, that really dictates where the symptoms are. The MRI patterns that we and others have shown, you can get involvement of the surface of the brain or the cortex, as well as the center of the brain, the deep nuclei. And you can see here in this case that we're showing that in our cohort, about 2/3 of patients have both the surface and the center of the brain involved. About 1/3 have only the surface of the brain, and about 2 to 5% only have the center of the brain involved. And for those from Mexico, the most common presentation in the E200K genetic form are patients who have the center involved, as well as a little bit of the surface shown in this figure here. And the sensitivity, the ability to detect these changes in CJD is very high, about 96%, and these are very specific. When you see these changes, they're almost always due to prion disease, but you do want to rule out other causes as well. Moving along now to acquired human prion disease. Kuru is the disease of the Fore ethnic group in Papua New Guinea, which is essentially extinct, though rarely cases occur because the incubation period after exposure to this disease is more than 5 decades. Iatrogenic forms of CJD, which are caused by medical or surgical procedures, can be divided at least into these 4 types or more I'll talk about, and then variant CJD, which is exposure to bovine spongiform encephalopathy prions, either directly from contact with contaminated beef or from people who themselves were exposed to contaminated beef, and then those prions were in their body, and then people were exposed to either their blood or to their brain tissue accidentally. So for Kuru, this is a prion disease of the Fore ethnic group in Papua New Guinea, which is just north of Australia. And Kuru in the Fore language means to shake or tremble. They practiced ritualistic endocannibalism, which means in order to honor their deceased relatives, they had these death feasts in which they would cook the bodies, but not in a way to destroy the prions, and then they would eat them. And it was mostly the women and children who had the parts of the bodies that had higher levels of prions in them. And the men tended to eat the muscle, which had less prions in them, whereas the women and children, unfortunately, had more of the brain and the spinal cord material. At the peak, about 1 to 2% of the population of the Fore ethnic group was suffered from this disease of Kuru. It was much more common in women than men, and it was much more common in older, in 20 to 40 years of age, in women to men as well. And it was a little less common in women, but still more common in women than men in those in younger people. And as I mentioned, the incubation period probably was anywhere from about 1 to at least 50 years. And when the cannibalistic practice stopped, the epidemic gradually died out. And here's a picture of Carlton Gajusek and colleagues in which they're examining a poor boy who is suffering from Kuru, and he's unable to walk in this picture. Now moving on to iatrogenic causes of CJD. These are types in which the disease is inadvertently caused by patients being exposed to prions through medical or surgical procedures, which is a really unfortunate form of CJD. But these forms have really decreased as we've become aware of them, and we've developed methods to avoid the contamination that would lead to spread of this disease. So on the top row, I divide them into iatrogenic causes into whether it was due to a surgical procedure or a medical procedure. And this precise method is in the second row. And the number of cases is the third row. And then how long it took for the disease after exposure until the person developed the disease. And you can see that the dura mater graphs, this is the dura mater or covering of the brain. And the covering of the brain, that tissue is very helpful and useful for medical procedures. And they would take, and still do take, the dura from people who've died, just people who weren't known to have prion disease. They took this dura mater and they used it in medical procedures after cleaning it and decontaminating it in ways to remove viruses and bacteria, but unknowingly they didn't remove prions. And about 228 cases, and mean incubation period is of about 12 years, but you can see there's a wide range in parentheses. There have been a few cases where neurosurgical instruments were used in one person, cleaned and sterilized, but again, not in a way that would destroy prions. And then these neurosurgical instruments were then used in other patients, and the disease transmitted from the first patient to the second patient, unfortunately. And the same thing with electroencephalogram or EEG depth electrodes. There were 2 cases in which they used to use very thin needles to actually measure seizures in patients, as opposed to now measuring the seizures on the scalp. It's much more sensitive to actually put a needle directly into the brain. And unfortunately, there were patients in whom they were measuring seizures who unknowingly had CJD, and then they cleaned and sterilized these needles for the next patients, not knowing that the first patients actually had prion disease, and the second patients that were exposed to those needles, unfortunately, the prions were transmitted to them. And then corneal transplants, and then medical procedures in which they take human growth hormone or gonadotropin. These are hormones taken from glands in the brain from patients who've died, and from people who've died, and they're extracted in mass from many people who've died, and then they're used to treat medical conditions such as short stature for growth hormone, or perhaps women or men who have either problems with gonadotrophins, particularly for women, trouble getting pregnant. And then there have been a few cases in which variant CJD, which I'll talk about later, in which there are about 4 cases now in which that was transmitted accidentally. Blood was taken from patients who unknowingly later developed variant CJD, and that blood was used and transfused the red blood cells into people. And those people who got the transfusions unfortunately developed variant CJD. The people who gave the blood didn't develop CJD until a couple years later. And at the time they gave blood, it wasn't known that they'd been exposed to BSE. So a typical variant CJD case we had at our center was a 20-year-old woman who was in the British military. She was in the Communications Corps. She developed some personality changes, depression, psychiatric changes. She developed ataxia or incoordination, then chorea. She came to our center in July of 2002, so about 9-10 months after onset. And then she came back 2 months later. When we saw her initially, she had difficulty. She had some cognitive and motor problems. 2 months later, everything had worsened, and she died about 3 months after her last visit with us. And she died from variant CJD. This was her MRI. And in this MRI, if you can see the bright white areas in the center of the brain, what we're showing there is the pulvinar sign, and that area is brighter than other parts of the brain. And that finding has high specificity and about 75% sensitivity for the variant form of CJD. So the MRI can be helpful diagnostically in this form. And here I point to it with these arrows. That's the part of the brain. Now, we can see that area bright in sporadic CJD, but usually that part is not the brightest part of the brain in sporadic CJD. And then on the diffusions, the top is a flare MRI, the bottom is a diffusion MRI, and you can see that it's also bright and even easier to detect on that MRI, perhaps. Variant CJD is different than sporadic CJD in several ways. Patients are younger, they live longer, their EEG and MRI are different, they have different genetic susceptibility. If you look at the brain under a microscope after a patient has passed away, we can see differences that can clearly diagnose and differentiate variant CJD from sporadic CJD. And the distribution of the prions is different. It's not only in the brain, it's also very prominent in the lymphoreticular tissue, which is why that form of the disease is easier to transmit. There have been no cases known in Mexico. There have been 4 cases in the US and 2 in Canada, but all of those cases were believed to have acquired the disease outside of the United States and outside of the West. On this slide, I'm showing the cases of mad cow disease or BSE in blue, and in green, the cases of variant CJD. And on the lower axis, I'm showing the year, and you can see that about 10 plus years after the peak of the BSE epidemic was the peak of the variant CJD epidemic. And there's good evidence linking the two. Exposure to BSE or bovine spongiform encephalopathy is what led to the human form of the disease. And here we're showing the cases by country. The higher the bar, the more the number of cases. Along the x-axis or the horizontal axis is the year. And as of 2023, there have been about 238 cases identified worldwide. And you can see that there have been almost no cases after 2016. There were 2 cases that occurred over the past 2 or 3 years in France, and those were from people who were working with the brains of patients who died from variant CJD, and unfortunately they were exposed. They weren't using proper precautions, and unfortunately they developed variant CJD from that. Now the last part of my talk will be about genetic prion disease. Here is a schematic of the prion protein gene, and in red are many of the mutations that occur. In blue are some of the novel mutations that we've identified at our center. In green below are some polymorphisms or variants that don't cause the disease, but in different populations can affect how the disease presents. And what I'd like to focus on is the codon 129 that hopefully you Dr. Knight spoke about, and this is a variant that can affect how the disease presents. It can affect the symptoms, it can affect the age of onset. And then for the G127V, the UK group in the UK showed that this polymorphism or variant actually is protective. And people in the Foray ethnic group who have this variant never developed prion disease. And when that research group developed a mouse model with this variant, you cannot give those animals prion disease, the mice models. So if we can actually use this to actually develop, we might be able to use this to develop treatments for prion disease. In this slide, this is a schematic of the prion protein gene from one of our papers from our group, and where we're showing in red and in blue these colored mutations are mutations that present or genetic mutations of prion disease that present fast, usually less than a year survival. In green are the forms that present more slowly, like Gerstmann-Strauss-Scheinker or GSS disease. And those can have survival generally of 3 to years to more than even a decade. And you can see that there are dozens of mutations in this prion protein gene that can cause prion disease. And we're studying many of these types in our center. The ones in purple above the line, those 4 or 5 or more, those actually have an X at the end, and those cause a form of the prion protein that's not the full length. It's truncated, shortened, and those present very slowly, very differently than other types. And then I'll talk, just to finish up the last few slides, I'll talk about the E200K form of genetic prion disease. This is the most common form of genetic prion disease worldwide. It's also the most common in Mexico and the United States. There are at least 4 geographical founders or origins of this mutation, including Sephardic Jews from the Mediterranean. Sephardic in Hebrew means Spanish. And the origin of this dates back to prior to the Inquisition, and most of the E200K families in Mexico and in Latin America seem to have this origin, where the families were initially Jewish. The other 3 are Slovakia, which is the second most common in the world, and then Japan has also an ethnic origin there, and then Austria-Hungary and Italy might have a common origin as well, a different founder. And the E200K presents with a lot of overlap with the classic sporadic form of CJD. And if you didn't do genetic testing, even if you had autopsy to look at the tissue under the microscope, you generally could not differentiate E200K from sporadic CJD, which is why we do genetic testing either from the blood while a person is alive or after they've died from the brain tissue in all of our cases of previously. And then another form of prion disease is called Gerstmann-Strauss-Scheinker, named after these 3 physicians who described some of the first cases about almost a century ago. And this is a typical case, though quite young onset. This is a 23-year-old woman with incoordination and memory loss. And these are her symptoms. She presented in March of 2004, and she died after about a 3-year course. And she had a mutation called A117V. She led a tough life, had a lot of drugs, didn't treat her body well. Her father developed the disease in the same month, the same year and month, but he was more than twice her age. And he died after about a 2-year course of the disease. And both had slightly different presentations. He was a little bit more behavior and cognitive, and she was more movement disorder. And this is their family tree. And what we're showing in black, the black round circle with the gray arrow, that's the 26-year-old. With the other black arrow, that's her father, who I'm sure she was 23 at the time, and he was 46. And then his brother is shown in black. And then in red are other family members who died of a disease that, even though they weren't genetically tested, we think they had the same disease. So this disease runs throughout the family, and as Dr. Knight said, they're all, this is generally an autosomal dominant disease. Just to briefly, at UCSF, we have a visit in which patients come in for 2 days. We do extensive testing on them, and we're looking for biomarkers in their early stage of disease. Here in this schematic, I'm showing time on the x-axis, and that vertical dotted gray line is the clinical onset of disease when symptoms begin. And the solid lines are biomarkers that we expect to see in people who have fast progressing disease, and those are the solid lines, and the dashed lines are the course of biomarkers, or either cognitive tests or CSF or blood or plasma biomarkers or changes on MRI that we expect to see and expect to differ between the fast and the slow forms of the disease. And what we're seeing here, where if you look at that blue arrow, in patients who, in their CSF biomarkers of fast, in fact, in E200K, we're finding some tests are abnormal as long as at least 4 years prior to onset of the disease. And the reason why that's important is that that means that we might be able to begin to treat patients with genetic prion disease as early as 4 years or earlier before the onset of the disease. So that's exciting. And there's also data suggesting that the MRI might also be positive prior, but by visual assessment, it's probably not as good as the one of the spinal fluid tests called the RT-QuIC that Dr. Inga Zur will talk about next, and Dr. Richard Knight briefly talked about. So at our center, we followed almost 200 patients who are at risk for genetic prion disease from 73 families, and we're bringing in people who have who are carriers, who have the mutation, and people as well in the families who are non-carriers. And they're important because they're our control population. We're looking for changes in people on the left, who those changes that are not present in people who don't carry the mutation, because those could be markers of the disease occurring, the beginning of the disease prior to clinical onset. And here we're just showing the number of families. So we're following 30 families, for example, with E200K mutation, shown on the very far left bar. And if we look at the individuals, we're following 59 patients who are carriers, and looks like 26 who are non-carriers with the E200K. And then the rest of the other mutations that we're following are shown to the right of that very far left bar. Well, just to conclude, I think human prion diseases are incredibly diverse. We'll talk more about the diagnosis. We think we can learn about the genetic prion diseases, can teach us a lot about how other forms of the disease occur, and we think that we might be able to treat, or more likely to be able to treat genetic prion disease even before the disease begins. And just for the sake of time, I think I'll skip the rest of my conclusions, but feel free to read these. I want to thank a lot of people, my collaborators at UCSF and other institutions, in particular our patients, our families, the National Prion Disease Pathology Surveillance Center in the U.S., the Center for Disease Control and Prevention in the U.S., and the various CJD support groups in the United States as well. And most of my funding has come from the National Institute of Health and from some families who have donated to my research. Thank you very much.
Ok. Em, Nada más para comentarles que estamos en una pequeña pausa. Reiniciamos en 5 minutos con la presentación de la doctora, la profesora Ingacer, con el tema de diagnóstico de las enfermedades por priones. Esperamos en 5 minutos. Muchas gracias. Hola, buenas tardes. Estamos de regreso para las transmisiones de este primer simposio de enfermedades por Priones aquí en México a través de la Fundación Fénix, la Universidad de Guadalajara, el Universidad de los Altos. Y es un gusto, un honor para nosotros tener con nosotros a la profesora, la doctora Inga Ser. La doctora Inga Ser es profesora de la Facultad de Medicina Clínica de la Universidad de Georg August de Göttingen, es jefe del Centro Nacional de Referencia de Enfermedades por Priones de Alemania y del Laboratorio de Neuroquímica del Departamento de Neurología. La doctora ha publicado bastantes publicaciones en el campo de enfermedades por priones y otras enfermedades neurodegenerativas, donde ha hecho contribuciones sustanciales a la investigación del líquido cefalorraquídeo y la imagenología en esas enfermedades. Su trabajo actual se centra en comprender la heterogeneidad clínico-patológica de las enfermedades crónicas en el desarrollo de biomarcadores fiables específicos de cada enfermedad. Sin más, le dejo el micrófono a la doctora profesora Inga Ser para escuchar su presentación sobre diagnósticos de enfermedades por priones. We're ready to start, Dr. Inga. Thank you so much, and we'll listen very attentively to you.
Yeah, thank you very much, Victor, for inviting me and also for being again in contact after so many years. And of course, COVID prevented meetings and everything, and we had a lot of problems. But I hope now we can overcome this and meet again one day. Okay, so I'm very happy that I was asked to talk about diagnosis of prion diseases, because this is, of course, my favorite topic, and I started working in the prion field more than 20 years ago, and I was interested in biomarkers. So this is how I came into the field. And just to remind you about the progress which has been made in the diagnosis of human prion diseases. Maybe you remember in the 70s and in the 80s of the last century, we virtually had nothing to diagnose the disease. Of course, it could be done by autopsy or by brain biopsy. This was the gold standard to diagnose the disease. And later in the 1980s, it became clear that there are some certain changes in the EEG, which are periodic, sharp, and slow wave complexes, which can support the clinical diagnosis. But of course, this was only one of the paraclinical tests and not very specific. Later, we had emerging biomarkers, for example, elevated levels of the protein 1433. Then at the turn of the century, we had realized that magnetic resonance imaging is important contribution. And now we are also working with direct detection of the misfolded prion protein. And we are still not done. We are now moving into the field of blood-based diagnostics. So this is the overall overview, and I would like to talk about some of these topics in a bit more detail. So for the biomarkers, there are different kinds of biomarkers which can be measured in different kinds of biofluids. For example, in the cerebrospinal fluid, we can measure abnormal concentrations of brain-derived proteins, such as 1433 protein, but we also know that tau protein, NFL, synuclein, GFAP, A-beta 4042, and so on are altered in the cerebrospinal fluid because probably of the general process of the rapid neurodegeneration. We also have some biomarkers which can be detected with novel techniques in blood, like NFL, tau, and the other kind of biomarkers which we have is related to protein characteristics, and these are the aggregation assays. So coming back to these biomarkers, as I said, we have elevated levels of 1433 protein, which was the first biomarker and was more or less specific for human prion diseases, and was also sometimes elevated in other conditions like after epileptic fits or after ischemia or inflammation. But these are conditions which, of course, can be excluded by appropriate clinical tests. And later on, we learned that other biomarkers are also altered in a similar way. But now, for example, for the magnetic resonance imaging, this is also a very, very useful investigation because we see abnormally high signal in the basal ganglia, in the thalamus, and in the cortical areas. And this is just to demonstrate. This is a case report of a female, 42 years old, disease onset, and she had memory deficits and cognitive decline, some manic symptoms and at onset. Then after 5 months, mild gait disturbances. After 9 months, it was already clear, severe dementia, ataxia, extrapyramidal symptoms. So the full-blown picture of the Creutzfeldt-Jakob disease. And you can see on these MRI imaging, these are diffusion-weighted images. And you can see the typical appearance of the MRI in patients with Creutzfeldt-Jakob disease. So we have elevated signal, so high signal in the basal ganglia, but we also can see high signal in the cortical areas. So this is the important test. And now the MRI is, of course, widely available. So sometimes it's really when it's done for dementia diagnosis and when you run appropriate sequences like DWI sequences, then this typical picture can occur. And there are only few other neurological conditions which look like these changes on the MRI. So this is really something very, very specific. Yeah, so then the next class of the diagnostic test is the detection of the aggregated or of the pathological form of the prion protein, and this is done by the aggregation assays. It is called RT-QuIC. RT-QuIC is the name for real-time induced quaking conversion. And it is based on the work which was first done by Claudio Soto, who did the protein misfolding amplification assay, which uses sonication. But the technique was later developed by Byron Corgi to the RT-QuIC methodology. And this is a robust technique which is now used worldwide for the detection of the abnormal prion protein. So how does it work? Maybe you can draw your attention on the second half of the figure. We have here we have a biological fluid where we assume that there are PRPSC seeds, so abnormal aggregates of the prion protein. So this can be cerebrospinal fluid, but of course also other biofluids. And we incubate this sample with recombinant prion protein. According to the prion theory, there is a direct interaction between the seeds, between the misfolded form and the recombinant form, and the recombinant form gets also the conformation of the misfolded form. So we get more seeds, we have an elongation of the seeds, and we get an aggregation. So we get fibrils. And these aggregations are broken down by high frequency shaking. This was something which was really revolutionary by Byron. So if you shake it with a high frequency, you get a fragmentation of these long fibrils, and you get new seeds for the conversion. So somehow this reaction works like PCR for nucleic acids, but it is for proteins. So with this repeated cycles of aggregation steps and shaking, fragmentation, and again conversion, aggregation, and so on, we can increase the concentration of our PRPSC seeds. And at the end, they can then be detected by fluorescence. And this is the signal what we see. We see in a sample where we have this we have the misfolded protein. At the end, we have a high fluorescence, and we get a curve which shows us that this is a pathological sample. So this is the principle for the ARTIC-QuIC technology. And this is just to show you that it is now used for more than 10 years in different laboratories. These are just some of the studies which use the ARTIC-QuIC to confirm the clinical diagnosis of Creutzfeldt-Jakob disease. So we have a lot of data now, and this was the reason why we could also go with a working group, go ahead and confirm or modify the clinical diagnostic criteria for sporadic Creutzfeldt-Jakob disease. And as you maybe know, that we had a clinical criteria of probable disease, which was based on the clinical symptoms like dementia, progressive neuropsychiatric syndrome, ataxia, myoclonus, extrapyramidal signs, and we needed to have at least 2 of them, plus either MRI, typical brain MRI, or positive CSF 14-33 test. So these were all criteria. And now we could amend them by the ARTIC-QuIC detection. And now we need only a progressive neuropsychiatric syndrome and a positive ARTIC-QuIC test to confirm the diagnosis. So it is nowadays much, much easier to achieve the diagnosis. And I strongly believe that we probably also do not need autopsies anymore, because we have this really nice technology which gives us very clear results for the diagnosis. So at that time point, we could also say, okay, we have now a biomarker, and this is a direct biomarker which shows us disease pathology. So do we have any open questions related to biomarkers? Is there anything to do in the field, or are we done and can do something else? And to answer this, I would like to show you this slide. This slide demonstrates the detection of the abnormal prion protein by either Western blot or by aggregation assay in various organs in humans. So as you can see, the PIP-SC is not only restricted to brain and spinal cord, but of course it was also shown to be in the olfactory mucosa. But there are some papers showing that it is also in the lung, in the spleen, in the liver, in the digestive system, and also in skin. So apparently it is spread throughout the body, and can be also detected in peripheral fluids. And this was our approach to see if we can find out if the test can be done in a more easily accessible biofluids. For example, lumbar puncture, of course, it is easy done, and neurologists were doing it. It has not so many side effects, but of course it can cause headache, local pain, and in a very, very rare circumstances, meningitis. This is one of the problems. The other one is that when we use cerebrospinal fluid ARTIC-QuIC, it is hard to apply for follow-ups and for therapeutic studies, because of course we will not do the lumbar puncture every 2 weeks. And therefore we were looking for other biofluids to see if we can improve the situation. So this is maybe you know them, Frank. You know them, Victor. This is Peter Hermann, Matthias Schmitz, and Susanna Correa. These 3 people worked on the project I would like to present. And Susanna was the technical head of it, and she developed the assay. So what we have done, we tried to look for the tear fluids. Why tear fluids? tear fluids are very easy to collect. You need just a strip of paper. It is frequently used in the ophthalmology to diagnose the dry eye. And yeah, it is easy to collect and can even be collected by a lay person. You do not need a physician or a nurse to do this. So this is something what can be done. So we collected tear fluids. And what Susanna has done is that she synthesized a specific prion protein which carries the E200K mutation, the human E200K mutation. She did also the analysis with other mutations. So she introduced in the protein also the mutation for FFI and also some others. But the best substrate which worked was the one which carried the E200K mutation. So this one was then chosen for further analysis. And I can skip this. And this is maybe the most important part of what I have to say today. It is we can detect abnormal prion protein using tear fluid. So in green you see detection, the quick detection in the cerebrospinal fluid in CJD patients, and in yellow these are controls, but in blue, the line in blue, these are tear fluids. And of course they are not giving that fast response as the cerebrospinal fluid, because of course the amount of the PRPSC is much less than in the cerebrospinal fluid, but it works. So we can detect abnormal prion protein in the tear fluid in patients with sporadic Creutzfeldt-Jakob. And of course, we tested a lot of controls for this. We had healthy individuals, most members of our lab. We had immune-mediated neurological disorders. We had Alzheimer's disease, Parkinson's disease, vascular cognitive impairment, and many others. So they all have been negative. So this is really, the detection was really specific in tear fluids and was only in Creutzfeldt-Jakob disease patients. So the sensitivity is not that high as in the cerebrospinal fluid. So normally we have in the cerebrospinal fluid, we have the sensitivity of around 90–95%, and in tear fluids is 75%. Yeah, so this is the sensitivity is lower. Sensitivity is lower, but this is something which can be improved on one hand, and also on the other hand, we have the advantages because we can collect tear fluids in a repeated way several times, you know. Okay, so, and then what we've done is we also had tear fluid samples from patients with different kind of mutations. So for example, mutation, the P102L mutation for Gerstmann-Streußler-Scheinker syndrome. This is on the slide. It is the picture number B. And it was also positive there in the genetic cases, so in GSS. But it was also positive, and this was very interesting, when we tested it for mutations, for patients with mutation, so with healthy mutation carriers, and in patients who were sick with a mutation. So for example, FFI, and so it was positive in patients with fetal familial insomnia, but it was also positive in healthy mutation carriers with fetal familial insomnia mutation, so presymptomatic. And also the other PRP mutations were also positive. So this is something which was very surprising and very interesting. And then, this is just, these are just the absolute numbers for the healthy mutation carriers, what you can see here. They are healthy at the moment. So we have P102L mutation carrier, we have FFI, and we have OPRI mutations, and also E200K mutation carriers. So these are, people at risk, if you would like to call it that way. And for the moment, we observe them for some of them for almost 4 to 5 years when we collected the first tear fluid. and they were positive in these tear fluids, but still negative, so no disease, so they still healthy. Yeah, but the test is already positive. And it seems also that it is positive when we look for those with the different mutations, when we had different time points from the same patient, from the same individual. And it seems to be positive, to get positive very early, and it stays positive. So it does not disappear and it does not change. So we had expected to have a negative result, for example, and a positive test close to the disease onset, but it seems not the case. It seems that the mutation carriers intrinsically have positive RT-QuIC. So how to interpret these findings? This is something which we really need to understand how this can be interpreted and how can we understand this. And when we look for other neurodegenerative diseases, we face a similar situation for the synuclein RT-QuIC. So this one is just these are just results from also from our laboratory on the synuclein RT-QuIC. So you can do the same procedure with other neurodegenerative proteins which are responsible for neurodegenerative diseases. And here these are just results for the synuclein RT-QuIC. So, and amazingly, we have a similar distribution as we see it in prion diseases. So abnormal seeding activity for alpha-synuclein has been detected in skin, in saliva, in olfactory mucosa, in the cerebrospinal fluid, blood, serum, duodenum, others. So it's also spread through the body. And there are very interesting papers available, which give us some clue. What does it mean to have a positive RT-QuIC test? For example, this is a study which is coming from Barcelona, from Alex Aranzo. He has an amazing cohort of patients with isolated rapid eye movement, sleep behavioral disorder as a prodromal or very early sign of Parkinson's disease. And he collected cerebrospinal fluid from these individuals and followed them for many, many years. And what he could show is that those who've been alpha-synuclein RT-QuIC positive at onset, not at onset, at enrollment, so no Parkinson's disease at that time, but positive RT-QuIC, they had a high risk to convert into clinical Parkinson's disease through the next 10 years. So almost 80% converted to the disease. So these are converters. So which means that you can be alpha-synuclein RT-QuIC positive very early, which is also maybe already a sign of the pathology, but it does not correlate with the clinic, but it tells you something about your risk. So, and a similar study has been done in Sweden, coming here from also in a similar setting, cognitively unimpaired individuals were followed also for 10 years, and those who have been positive at enrollment converted to Parkinson's disease or to Lewy body disease after 10 years. So these are the studies. So what we need to understand is that apparently we have different kind of biomarkers. We have at-risk prodromal biomarkers, which might be something like the RT-QuIC prion protein or the RT-QuIC synuclein, or maybe also other neurodegenerative diseases. So they are more or less at-risk prodromal biomarkers. And of course, sometimes there are also diagnostic biomarkers, but they are different. And we need to understand this, for example, in healthy mutation carriers, that in healthy mutation carriers, we have at-risk biomarker, but this is not a diagnostic biomarker in the clinical situation. Yeah. So, and what does it mean for the moment? It means that we also can search further for biomarkers which are probably more capable to tell us about the disease. So when the disease starts, and this is not PSC, these are other biomarkers. For example, the NFL, this is neurofilament light chain. And it has been studied extensively for many, many neurological disorders. It started with motor neuron diseases, but also for other diseases. Has been shown that it is an important biomarker. But most of interesting is it is a dynamic biomarker, so it changes with disease progression. And here, what we have done here is we analyzed plasma NFL levels in patients with fetal familial insomnia and controls, and you can see there are clear differences in plasma for this biomarker in clinically affected patients. And then we had one individual who came to our clinic. He was a mutation carrier for FFI, and he gave blood for several times. And after he got sick, we analyzed all these samples we had from him, the plasma samples. And what we can see here is that it seems that the NFL levels start to increase at disease onset. But disease onset at that time was something which was really hard to diagnose, because he had depression, he had some sleep disturbances, and it was not very clear, is it now FFI starting, or is this kind of a psychological reaction to the disease? But the biomarker was already increasing. So apparently this could be one of the potentials to diagnose the disease, or to at least to help in the disease monitoring. And there are similar data were obtained from UK. So Simon Mead, he also followed several mutation carriers at the presymptomatic stage, and also the converters. And he could also demonstrate that NFL is one of the parameters which might start to increase very around the clinical onset of the disease, not before, but it might be a biological parameter which tells us that the pathology is now going on. So what we still need is, of course, we have biomarkers, we have now at-risk biomarkers, we have now diagnostic biomarkers. But for example, for disease monitoring, for clinical trials, once we will have clinical trials, I'm pretty convinced that there will be some progress in the future, in near future. So here we need a biomarker which is dynamic and which tells us about something about the neurodegeneration. So the time of the biomarkers is not over. There is still a lot of to do and to understand what we are dealing with. And with this, and this is what I also wanted to mention, that on the last Prion 23 meeting in Porto, we had a meeting of people who are doing the prospective clinical studies in healthy mutation carriers. And because I realized that a lot of activities going on worldwide, and there are many people expressing interest to join forces for that. So we created a group which is called Prion at Risk. And yeah, so we are very optimistic that we can put data together and yeah, join forces for the biomarker development and detection in this field. Yeah, just to show you the people. And these are people from my laboratory who, of course, have done this work I presented to you, and many of them are working as clinicians, but there are also people in the laboratory. There are people who are dedicated their work to biomarkers, and yeah, these are people I just need to thank them that they support me and our activities in that. And with this, I would like to stop. Thank you for listening, and I don't know if there are questions allowed or not, but thank you very much for listening. Thank you.
Thank you so much, Inga, for this magnificent presentation. I could get track of those publications at the New England that you showed to us today, but I didn't know some of the rest of the publication that you showed. I usually tell my students that the eye is part of the nervous system that you could easily look at. So, and I guess this study that you did on the tears of the patients is very useful to have an easier way to access the nervous systems, or eventually when the sensitivity of the test it goes up a little bit higher, maybe this could also be one very useful way also to diagnose any prion disease. I, I I don't know if there is any question from the audience. I guess we have a few minutes for that. Maybe one for at least one question. And in the meantime, I will say that I remember Matthias. So if he's still there, then he's been there for a while. It is good
Yes.
to see him in your group. Uh, Inga, have you tried doing all of this testing, because you showed some studies that you did with Alzheimer's and Parkinson's? Have you done any specific studies focusing on any of these neurodegenerative diseases, as like Alzheimer's or Parkinson's, as the way you did for CJD? I mean, using them as the target of the diagnosis, but not as controls for any other neurodegeneration.
Yeah, of course, we now are very encouraged by the data from the prime protein in tear fluids. And we also have done some studies on tear fluids in Parkinson's disease, for example, and in Alzheimer's disease. And it's really amazing, because in tear fluids, it is a very clear, how to say, fluid, biofluid. You know, there is no blood and there are not a lot of cells which distribute the reaction. And this is a very, very easy to collect. And it's a very good platform for any kind of biomarkers. And what we have done, I can tell you, we have done some tau and A-beta in Alzheimer's disease. It works quite well. And we have done some work on Parkinson's disease, and we measured. I don't remember, but it was difficult for Parkinson's disease because there is not really a clear biomarker. But it is detectable. You know, we can measure synuclein in tear fluids in Parkinson's patients, but it is not diagnostic. Yeah.
Okay,
but
good.
Of course, yeah, we are working on it to have the RT-QuIC done in tear fluids, also in synucleinopathies.
Yeah, there are some other publications concerning Alzheimer's, where there is autosomal dominant forms of the disease, and mutation carriers who haven't developed the disease. They've done some studies looking for biomarkers in the preclinical stages. So maybe this tier test that you've been doing can also be applied to this population in autosomal dominant. So as they are carriers of the mutation, then maybe you can track how this biomarker is developing in the preclinical stage, because it's been done in CSF, it's been done in blood, obviously, but I believe tears is also a very good way of looking at the
and the most important issue is that it is so easy to collect, so you don't even need a nurse or a physician. So this can be really done by somebody. So this is really not complicated to do it. And I think this could be, for clinical studies, this could be a good platform for biomarker detection.
Yes, and I may have misunderstood, but in assessing the tier test, did you use CSF as a gold standard just to compare? Okay. Okay.
Yeah, so it's still CSF is still our gold standard for the diagnosis, and this tear fluid stuff is very new. So we don't, for the moment, we don't know if it can replace or not. Probably it cannot replace CSF because, of course, you are doing cerebrospinal fluid at least once to exclude inflammatory diseases or whatever. This is the main reason, the autoimmune mediated encephalitis, which is an important differential diagnosis. So at least one cerebrospinal fluid is needed. But if you want to do some kind of monitoring or for clinical studies or whatever, then you cannot go with the CSF. You need to take something else.
Yeah, I got one question that I also had, is that could you correlate these changes in the tear fluids with changes in the retina? I mean,
No, we have not done that.
Okay,
Yeah.
okay.
We have not done that because we also don't have any material to do this.
Oh, okay. Okay, good. Well, yeah, I guess we had a very good. I mean, I know that we have a very good talk and presentation. We at FENMEX, the Mexican for the Degenerative Diseases and Prion Diseases, would like to thank you for your presentation. I guess we had the diploma for your presentation. It's been shown at the YouTube presentation. We will obviously send it to you on email. We would like to thank you again for your talk, and we will move forward for our next speaker, who is Eric Winikel. And Inga, I'm very happy, delighted, and honored to have you with us and to accept our invitation in a very quick way, because I emailed you on Saturday, and I know it was the evening in Europe, and you immediately replied in a very positive way. I'm very happy and very honored. Thank you so much, and we do hope you to see you soon and to have you in Mexico, hopefully next year.
Okay, thank you very much, Victor. So have a nice meeting again, and good luck. Thank
Okay,
you.
see you.
Bye.
Goodbye. Bye. Okay. Eric, you're here with us.
Hi, how you doing?
I'm okay, thanks. Just let me ask a technical question. ¿Estamos siendo transmitidos, Emanuel? ¿O hay una pausa? Una pausa, por favor, sí, en la transmisión muy rápido. Gracias. ¿Listo? Buenas tardes, aquí estamos de nuevo dando continuidad con nuestro simposio de enfermedades por priones. Hemos llegado ya al tratamiento de las enfermedades por priones y está con nosotros el doctor Eric Minike. Nos da mucho gusto que también haya aceptado nuestra invitación. Es un honor tenerlo aquí en nuestro congreso. El doctor Eric tiene una licenciatura en Lengua y Literatura China de la Universidad de Wisconsin en Madison, donde estudió una maestría en el Instituto de Tecnología de Massachusetts y posteriormente un doctorado en Ciencias Biológicas y Biomédicas por la Universidad de Harvard. El doctor Miniker se formó en los laboratorios Broad a cargo del doctor Neil MacArthur y Stuart Schreiber. Codirige la iniciativa para desarrollar medicamentos preventivos para enfermedades crónicas en el Broad Institute. En enfermedades por priones, su investigación abarca el descubrimiento terapéutico, el desarrollo preclínico, la investigación de biomarcadores clínicos y de la historia natural de la enfermedad. También estudia la genética de los priones y otras enfermedades neurodegenerativas y la aplicación de conjuntos de datos genómicos humanos a la epidemiología de enfermedades y la validación de objetivos farmacológicos. Tiene un blog, cureffi.org, el cual cubre temas que van desde la biología de los priones hasta la genómica y el desarrollo de los fármacos. Sin más, le dejamos la voz al doctor Eric Minickel, que se encuentra con nosotros, y estaremos escuchando su presentación para posteriormente abordarlo brevemente con algunas preguntas. Muchas gracias y escuchamos la ponencia del doctor Eric. We'll start in a moment with
Hola,
your video.
yo soy Eric y
Again,
soy
thank
un
you for being here
científico
with us.
aquí en
And
Boston.
we're
Quería
starting
presentarme
right now.
un poquito brevemente en español, pero porque hace muchos años no he hablado español frecuentemente, haré la mayoría de la presentación en inglés. Discúlpame, Víctor me dice que él y sus colegas le van a traducirle. Uh, Uh, yo soy un científico aquí en el Broad Institute de
Parece que no se
Harvard y
escucha el
MIT.
audio.
Y yo trabajo juntos con mi esposa Sonia. Y juntos dirigimos el laboratorio con el solo objetivo de desarrollar un tratamiento eficaz para la enfermedad priónica durante nuestras vidas. Mi esposa corre riesgo de esta enfermedad porque ella heredó una mutación de su madre, quien murió a causa de esta enfermedad en 2010. Cambiamos nuestras carreras para dedicar nuestras vidas a la cura. So thank you for bearing with me with my terrible Spanish. I thought it would be nice to do just a short intro. The rest of the talk I'll give in English. So this is Sonia and me on our wedding day in 2009. Her mom organized the whole wedding and was, in some ways the star of the show. This is her helping Sonia get ready that day. It was only 6 months after this photo was taken that she got sick. And as I'm sure everyone in the audience is familiar, the disease is so rapid, you don't even know what's happening, right? Her first symptom was blurry vision, weight loss. A few weeks later, she couldn't finish a sentence. A few weeks after that, she didn't recognize her family members. Soon she was permanently hospitalized. So devastatingly rapid neurological decline. And the whole time, we had no idea what was happening. It was only from her autopsy that we found out that she had died of prion disease and that it was genetic. So the fact that it was genetic meant that my wife, Sonia, was at 50-50 risk. Many people told us not to get genetic testing because they said, there's nothing you could do. Why would you want to know? But we're the kind of people, we just had to know. We had to know if she had the mutation. And so we got genetic testing, and it was bad news. Sonia did inherit the mutation. It's the D178N mutation from her mom. And this means that Sonia is very likely to develop the same fatal, untreatable neurodegenerative disease that her mom developed. And that changed our lives. So from getting that genetic test report, we quit our jobs. I had trained as an engineer. Sonia had studied in law school. We quit our jobs and we got jobs doing scientific research at Massachusetts General Hospital. Eventually, we went back to earn our PhD in biomedicine from Harvard. And in 2019, we defended our PhDs. And since then, we run a lab here at the Broad Institute where I'm sitting. It's a joint research institute of MIT and Harvard. And we have 12 people in our lab now. And the entire focus, what everyone works on, is developing a cure for prion disease. So this is us in the laboratory. And one of the first things that we had to think about is finding the true name of the disease. Because when Sonia's mom got her autopsy report, when Sonia was given her genetic diagnosis, we were told that the disease was fatal familial insomnia. And you'll hear many other names for different subtypes of prion disease. And the key insight we've had is that these are all one disease. Even though you hear many different names, it is all one disease. It's called prion disease. And the reason we can call it one disease is that it's all caused by the same molecular mechanism. It's all caused by one protein called PRP. So every human has this protein in their bodies and in their brains, and normally it's not a problem. But it's capable of changing shape. So the same protein, it just changes shape, and now you've got a bad shape. And what that shape does is it goes around and grabs more copies of the protein and turns them into more of that bad shape. And so it spreads exponentially across the brain and it kills neurons. And for many years, people thought prion disease was mysterious because how could a protein copy itself? But actually, it fits squarely within everything we know about biology. Your genes are DNA. They give rise to an RNA that's the messenger that carries the code in that DNA to be made into protein. Proteins are the machines that actually do the work of the cell. That's all normal. The only difference in prion disease is that after you have the protein, then it can misfold, and the misfolded state can propagate itself and cause disease. And the solution to this, we think, is simply to make there be less of that protein in the first place. And I'm going to talk through 5 different puzzle pieces that we think are important to bringing this to be a reality of treating prion disease in our lifetime. So first is understanding the genetics of this disease. And for each of these puzzle pieces, I'll just spend a couple of minutes on 1 or 2 research studies that we've done. So one of our first questions was, which mutations in this gene, the prion protein gene called PRNP, actually give people a high risk of disease? Because there were something like 70 different mutations that have been seen in patients with prion disease. But for many of them, we just didn't have evidence. Does that mutation cause the disease, or is it there as a coincidence? And so we did a study 7 years ago now, in which we found that the mutations are not all equal. So unfortunately, the bad news for Sonia is that her mutation, D178N, really does seem to give you a very high risk of disease. It's probably not 100%, but it might be 90% risk of disease in your lifetime. However, there are other mutations that seem to cause a much lower risk. And I'll just note that one of these is T188R, which I know has been seen in several families in Mexico. We believe that that mutation increases risk of prion disease compared to the general population, but that in numerical terms, it's still a relatively low risk, and that a person with that mutation will probably not die of prion disease. Another study that we did was, when will disease happen? So given that Sonia has this mutation, how much time do we have to develop a cure? And we gathered data from 9 different study centers worldwide on over 1,000 patients. And what we found is that there's a huge variability in when people get disease. So for Sonia's mutation, the average age is about 51, but the range has been from 12 to more than 89 years old. And this is true to varying degrees for every mutation. There may be an average, but there's a huge range around that. And so far, we don't have anything that can predict where you might fall within that range if you have the genetic form of the disease. In particular, it is not predicted by parents' age of onset, as far as we can see. So the important thing about these two studies, I think it's a value for people who are at genetic risk of disease. Another implication is that it helps us to identify who are the people with a very high disease risk, because maybe that's a population in which we can think about using a drug to prevent the disease. And so next, I'm going to talk about our animal studies in which we've tried to develop a therapeutic for prion disease. We did some studies using an antisense oligonucleotide, which I'll introduce in just a moment. And the data that I'll show here is from what we call a tool compound, meaning it's just a proof of concept that we can use in a mouse, but it targets the sequence of the RNA in the mouse, and that sequence is different in a human, but we can also make a sequence for the human. And so this is sort of a test case to see, does the idea of the drug work? And the idea is that this leads to a human drug down the road. First, I want to say briefly how we study prion disease in animals. So not only humans get prion disease, we see prion disease naturally occurring in goats and sheep, cows, deer, elk, camels. Many different animals get prion disease. And when we inject prions into the brain of a mouse, that mouse will develop a fatal disease 5 months later. And that fatal disease looks exactly like the human disease. So the symptoms that the mice get are very similar. When people cut open their brains and look at the protein deposits in the brain, it's very similar. I would say this isn't a model so much. It really is the true disease in a mouse. And the drug type that we wanted to test in these mice is an antisense oligonucleotide. We call it an ASO for short. An ASO is a short sequence of chemically modified DNA. So this example is 20 DNA letters, and it's reverse complement, which means upside down and backwards, so that it binds the same way our 2 strands of DNA bind to each other. This piece of DNA can bind to the RNA that carries the message for the prion protein gene. And when those two bind, it causes an enzyme that we all have in our cells called RNase H to come and cut the RNA. And when it cuts the RNA, it can't make as much protein. So now we're lowering the amount of prion protein that's produced. And here was the really exciting result that we got. So when we infect mice with prions, they develop fatal disease after about 150 days, 5 months. So on the x-axis, you're looking at how much time since the animal was infected. And on the y-axis, you're looking at how many of the mice are still alive. So you can see that after about 5 months, the mice die really all at the same time. Within about a week of each other, all of those mice will die of prion disease. But when we gave them the ASO, we see the long blue dashes. Every 90 days, we give them another dose, and this lowers their prion protein in the brain by about 50%. It made them live 3 times as long. The longest lived mouse was almost 500 days. This is similar to, in the red dashes, if we delete 1 copy of the gene, which is another way to lower the protein by 50%, they also live about 3 times as long. You'll notice here that when we treated the animals later, so these dots is if we waited until the animals were almost symptomatic and then we gave the ASO. We still increased their survival, but not by as much. So this made us think that in any patient with prion disease, this strategy of lowering PRP should help. An ASO could be an effective drug, but it's really, really effective when we can reach people early, when we can reach them before they're sick, if they're at genetic risk. So this motivated us to think not only about how can we advance this drug to do trials in patients who are already sick, but also can we find a way to do trials for prevention in people who are at risk. So this brings me to the next 2 points. I'll talk very quickly about a biomarker and then about natural history of people at risk. So one of the questions when you develop a drug, clinical trials take years and years. And sometimes it's too long to wait to say, I treat a patient with a drug and then I have to wait and see how long they live. And then maybe I have to do that in 100 patients because how long the patients are going to live is variable. And I need statistical power to say how long will they live. So it's really nice to have a quick molecular measurement to ask, is the drug even doing its job? And here we're lucky because the ASO lowers the amount of PRP in the brain, and your brain is surrounded by cerebrospinal fluid, CSF for short, which is this fluid that you can also measure by a lumbar puncture, so a needle into the spine. And for some people that sounds scary, but I have undergone a lumbar puncture 6 times, and I have to say it's fine. I don't think it's worse than getting my blood drawn. And so we can go into someone's spine and take out 20 milliliters of CSF, and we can measure prion protein in it. And I'll just show one data slide here, which is that we modeled this in rats. We treated rats with an ASO, and we showed on the x-axis, we lowered PRP in their brain. The higher dose we go, the more we lower the protein. And then when we measure it in cerebrospinal fluid, in CSF, we also lowered the protein. So this suggests that if we take this drug into a human and we measure the prion protein in their spinal fluid, we can guess how well the drug is working in their brain. So that's important because this gives us a way, even in a person who's not yet sick, and so we can't ask, did they get better? We can ask, is the drug doing what it's supposed to do? So what about those people who aren't sick? I showed you earlier that we've identified the mutations that seem to cause a high risk of developing disease, and we know to some extent at what age people might develop disease. But what's going on all those years beforehand? If someone doesn't get sick until age 50, what was going on in their brain those 50 years? You're born with the mutation. You have the mutation your whole life. Why is it that it's not until age 50 that you get sick? So we started recruiting people at risk for genetic prion disease, people like Sonia, to come to Boston and participate in a clinical research study, meaning we're not giving people a drug, but we're taking blood and spinal fluid and we're doing measurements on people to try to understand the disease process. In the 6 years since then, we've had over 60 people come, mostly from the US, but a few from other countries. The study's still recruiting. You're welcome to look at this link and learn more about it. And I'll just share 2 brief pieces of data. One thing that we measured is a biomarker called plasma NFL. So this is in blood. We can measure a little piece of a protein that comes from dying neurons. And so in patients who are sick, this protein goes way up in the blood because their brain is under a lot of stress, and that protein is leaking out into the spinal fluid and then into the blood. When we looked in people who are at risk but aren't yet sick, we did not find that that protein was elevated. That suggests to us that at least in most of these people, most of the time, their neurons are not yet dying or not yet distressed. The disease process doesn't seem underway. Even better evidence for that is that we can measure prion seeds. So the disease is caused by this misfolded protein. We can measure that misfolded protein in CSF. And in symptomatic patients, shown here in red, almost every patient is positive by RT-QuIC, which is this spinal fluid test. So almost every patient has detectable prion seeds in their spinal fluid. But when we look at people at risk who aren't yet sick, we only saw those seeds in 1 out of 23 people we looked at. Again, this suggests that for most of those 50 years, someone is totally normal. It's not as though you have the disease going on for decades and then it finally leads to symptoms. We think that people are totally normal for decades. Then the disease process starts, we still don't know why, and from then it could only be a year or a few years until it manifests in symptoms. The good news from all of this is it means we might have an opportunity to truly prevent the disease. If we look at people at risk like Sonia, if the disease process hasn't even started yet, then we can treat them before prion protein even begins to misfold. And maybe that's a really golden opportunity to do a lot of good. Just like I showed you in those mouse studies, that early treatment can triple survival, where a later treatment only gives you a smaller benefit. In order to make the idea of prevention of prion disease a reality, we also need to talk to regulators. So here in the US, that's the FDA. And what regulators are used to seeing is most drugs are tested in patients who are already sick, and the goal is to make them decline a little bit slower, or if you're really ambitious, maybe to stop them from getting sicker. And this is a different idea. We're saying, can we treat people while they're still healthy, keep them from ever getting sick? There's not that many precedents for that, but there are some. And one example is the cholesterol lowering drugs that I and millions of other people take to keep our cholesterol low to prevent a heart attack. If you take these drugs in the moment when you're having a heart attack, they don't do any good. It's too late. You need to prevent. And so there is a precedent in regulators' minds for the idea that sometimes we need to do clinical trials. We need to take an experimental drug into people who are still healthy to prevent them from later getting disease. The question in those trials becomes, what do you measure to show that the drug worked? So we went down to FDA, and I'm showing here some pictures from our first meeting, but we've talked to them a few different times now. And we've been really happy to hear that they are supportive of preventive trials, and they're open to the idea that we can measure CSF PRP. So I told you that in the rat, if we lower PRP in the brain with an ASO, we can measure that in spinal fluid and show that the drug did its job. We asked them if we could do that in a human who's at risk for prion disease and show that we lowered the protein in their brain by measuring it in spinal fluid. Could that be a way that a drug could get provisional approval? And they said, here's a lot of work to do, but yes, in principle, that would make sense. And this is a way that you could do preventive trials. So Sonia and I have our work cut out for us. This is why we have 12 people in our lab. We have a lot of work to do, gathering all the evidence that PRP lowering will be helpful, finding ways to lower PRP more and more potently and do it safely, and then measure and prove that we did that. So there's a lot of pieces that come together to make a good clinical trial and a good plan for a drug development, but we're working on it. One piece of that is finding the people. And we've launched a website, prionregistry.org, where people who are at risk can sign up. Part of why you sign up is to stand and be counted. It helps us because when we go to funding agencies or pharmaceutical companies or regulators, and we say we want to do preventive trials, they ask, well, do you even have the patients? Do you have enough people to do it? And if we can point to this and say, yes, we have hundreds of people who want to participate in these trials, that helps us a lot. This is also a way that you can volunteer for research and find out about research studies that are enrolling, such as the study in Boston that I told you about earlier. So looking ahead at what's to come, the ASO that we studied in mice, that project is partnered with Ionis Pharmaceuticals, a drug company based in California. They announced last month that they hope they'll do their first in human clinical trials sometime in the next few months. I don't know if that means next month or early in 2024, but they say that it's, quotes, very soon. Remember that a clinical trial is still just an experiment. Just because a drug has been tested in a human doesn't mean it will work. In fact, a majority of drugs fail. And so that's why we need to be testing additional PRP lowering drugs and finding ever more ways to lower this protein because we understand this disease. We know what we need to do to treat it. It's just a matter of finding a drug that is safe enough and potent enough to do the job. And the hope is that through all these puzzle pieces, understanding the genetics, having a biomarker to measure the effect of the drug, confirming our therapeutic hypothesis in mice, having a patient base of at-risk patients who could participate in prevention trials. The hope is that all of these pieces will be relevant, whether it's for this trial or for the next drug that comes along. We believe that we can do this in our lifetimes. And I'm sorry that we're not there yet. I know that for people who already have a sick loved one today, none of this comes soon enough, but we are working on it, and it is our life's work to try to develop an effective treatment for these diseases. Thank you so much for listening.
But that you also made it as a goal in your career, and I guess it's giving a lot of results and great results. One question: Eric, I know that by going all the way to the mouse models and then into humans, it takes some time. How long do you think that a viable drug would be available following your studies?
It's a very good question. So I think that we will see a first in human trial of a drug probably within a few months. Ionis originally said by end of the year. I think it may slip a little bit, but I think it seems very soon. But from first in human trial, most drugs don't succeed. If a drug succeeds, now first I'll tell you something very pessimistic, and then I'll give the slightly more optimistic tune. So on average, in all of the drug industry, it's about 11 years from first in human dose until drug approval. So that makes it seem like we have a very long time to wait, and only about 1 in 10 drugs will succeed in those 11 years. However, I think we have a very rapid disease, and we also have a very rare disease. And I think that those factors tend to mean a quicker development plan. So it could be that from first dose in human, if the drug works, the approval could be only maybe 5 years away. And I like to think that our probability of success is greater than the usual 10%, because we're very certain that prion protein is the right target. And many other failures we've seen recently, for example, in Huntington's disease, in ALS, I don't know for sure, but many scientists debate, were those failures that the drug did the wrong thing, right? It's not that the drug was too toxic. It maybe was they had the wrong idea about how to treat the disease, the wrong therapeutic hypothesis. So if you accept that we're very likely to be correct about therapeutic hypothesis in prion disease, I think we could have a better probability.
Right. Right. Thanks, Eric.
Yeah.
How do you see the future of prion diseases treatments following this or any other therapeutic approaches? We know that Ionis, you already said that it's about to start the clinical trial. This drug that you helped to develop is also starting another clinical trial. I was invited to participate in there because of you, for which I am very grateful for that. But what about the rest of the therapeutic approaches for prions? Knowing that you are very into this field, what are your expectations on overall for the therapeutics on prion diseases?
For the long term, I'm very optimistic. I think the hard thing is talking to people who have a family member who's sick right now, because it's such a rapid disease that if someone's already sick, we just can't get there in time. But if I look at the next 5 years or 10 years, I think that we should have multiple chances to make an impact on this disease. The ASO is just the first. If you look at other diseases where an ASO has been helpful, I'll give the example of spinal muscular atrophy. Spinal muscular atrophy was considered untreatable, uniformly fatal disease just a decade ago. And now, first there was an ASO in 2016, and now there's 3 approved drugs. There's an ASO, a gene therapy, and a small molecule. And last I checked, I think there's between 10 and 20 other drugs in development for spinal muscular atrophy. So I think that part of what the ASO gives us is the ability to prove the therapeutic hypothesis, to test what the design of a clinical trial could look like, find the patients, motivate neurologists to try to diagnose the disease earlier. And all of those are assets that will make other drugs come along. It just takes time.
Thanks, Erika. I don't know if we have another question from the audience. I want all of you to know that this date, and we didn't talk about it at the beginning, is a very a specific date in the United States, because it's practically Thanksgiving, and we also would like to double thank you, Eric, for connecting to our meeting, because we know it's a very familiar date, and even though that you are gathered with your family, you still made some time to be with us, to be at this presentation, to share your history, your progress, and to answer some of the questions. So there are some other questions that because of the time I will pass them to you, and then maybe by email or after this. I know you are very able to understand. I mean to answer. And again, we thank you for your presentation, Eric, for your willingness. And we've been seeing you every year at the meetings here and there. And when I invited you on July, you were very happy to share your story with us and your efforts. Thank you again. And we will move now to our next speakers.
Thank you.
Who are here with us. Thank you, Eric. Who are here with us, and I don't know if we're still seguimos transmitiendo, Emanuel, o tenemos una pequeña pausa. Hola, estamos de regreso con ustedes para nuestra penúltima charla del día de hoy, que se llama Las sesiones de pacientes en apoyo de los pacientes familiares del CJD. Se encuentra con nosotros Susan Solvins, quien es directora y coordinadora nacional de la red de grupos de apoyo para la enfermedad de Creutzfeldt-Jakob, cuyas siglas podemos ver o veremos en la presentación en un momento, CJD-ISA. Ella se convirtió en fundadora de esta red en 1993 como coordinadora de Nueva Gales del Sur y en 2004 fue nombrada directora. Ha jugado un papel importante en la expansión de las redes para ofrecer apoyo y asistencia a todos los australianos afectados con enfermedades por priones. Ella se encuentra en Australia, es actualmente la encargada de la red de apoyo para enfermedades por priones en Australia. Y cuando forma la alianza en 2006, asume el papel de copresidente de la alianza, un papel que todavía brinda la oportunidad de trabajar con organizaciones similares en todo el mundo. Y ha formado una red de Friends and Advisors, amigos y consejeros de las enfermedades por priones a nivel mundial. Susan recibió en el 2019 la medalla de la Orden de Australia en la lista de honor del cumpleaños de la reina británica por su compromiso con la salud de la comunidad. Y también se encuentra con nosotros Dina Simpson. Es también en inglés es co-chair, es la codirectora precisamente de esta red internacional de enfermedades por priones, el CID International Support Alliance, enfocada a apoyar a los pacientes de las familias que han estado impactadas por alguna enfermedad por priones. Ha apoyado también a varias organizaciones en varios países para establecer las redes de apoyo a las familias con enfermedades por Priones y también ha apoyado y ayudado a formar este grupo de Friends and Advisors a nivel internacional. Sin más, le dejaré la voz a Dina y a Susan para para que hablen con nosotros sobre las redes de apoyo internacionales. We're ready to hear you, Dina and Susan, and thank you for being here with us.
Thank you. I'm Suzanne Solvins, the director of the CJD Support Group Network in Australia. I'm a founding member and director of the CJD International Support Alliance. And I'm passing over to Dina, who's going to tell you about our work today.
Good afternoon, Ola. My name is Dina Simpson. I am a nurse by background. I come from a genetic impacted family of prion disease. We've lost about 22 plus family members over 7 generations to CJD. I started CJD Insight after my mom's passing in 1998. And I'm also the co-chair with Suzanne of the CJD International Support Alliance. We're very happy to be here today. We're going to share a little bit about the work of the Alliance, how we collaborate and assist other countries with their support organizations. And hopefully you'll learn a little bit more about us and what we can do collaboratively across the globe. So in 2006, we were at the CJD Foundation Conference in the United States in Washington, D.C. And Suzanne, a visionary, realized that there would be a positive outcome if we were to join forces of the support organizations that existed at the time for prion disease to come together and have a unified voice around the globe and leverage each other's expertise to really impact how we can support those impacted by prion disease. So this is a picture taken in the lobby of the Washington Court Hotel, where we pulled together Suzanne from Australia and her team, 2 of the support organizations in the United States, Japan and the United Kingdom, and we formed the Alliance at that time. We have 5 key objectives that I want to just highlight for you today. First and foremost, we want to be able to assist other countries in establishing their own support organizations in their native country, or to help the existing support organizations expand their services. Our focus is really on how we can support the patients and families, the caregivers, and those at risk of prion disease is one of our primary objectives. The second is to really promote continued access to care for those that are in active disease or those at risk. There's a lot of stigma that goes around with this disease, a lack of awareness on the part of healthcare professionals and others across the entire care continuum. And so it behooves us to really advocate for those that are impacted and make sure they have access to care, that we provide education to healthcare professionals and others so that people with prion disease are not dismissed, that they actually get the care they need to go through the process. And then for genetic families, after they've lost someone, there's other issues and things that we have to deal with on an ongoing basis that requires support as well. The third is really around supporting research efforts. We are not researchers, but we support research. We know how important it is for us to help find a treatment or cure. So that is one of our key objectives as well. Fourth is around providing avenues for patient and families to really work with pharmaceutical companies, academia, and other medical professionals. As we talk about and heard, as you heard from Eric, potential research clinical trials coming up, therapy options, and really leveraging the organizations in countries where they exist. So we can be that conduit to find patients to participate, educate families about what it means to be in a clinical trial, and really facilitate that connection. And the last is really to continue to advocate for awareness and the needs of those that are impacted by prion disease across the care continuum. And when I talk about the care continuum, it's not just the medical care continuum in a hospital or a doctor's office, but it goes beyond that. So hospice care, educating them and bringing awareness to them and how it's different in treating those components of the disease till it really becomes about end-of-life care, palliative care, funeral home education is critical as well. There's horrific stories that we've heard from patients or from families about the lack of getting what they needed for their loved one as far as funeral services, et cetera. So just to give you an update on where we stand today as an organization. Since we formed in 2006, we've added 9 new member organizations that you can see here on the left side of your screen. Those are full members, and they're at varying stages in their what services they offer and their growth within their own company, and we'll talk about that in a little bit. Then we have associate member organizations. These are countries that have someone has stepped forward and said, we want to start a support organization in their native country. So they're in the beginning stages, and we're here to help them through that process. And then we have a new group that we've added recently called Country Contacts. It's important for families to connect with others who have experience with the disease, other family members. It's so powerful to talk to someone who knows what you're going through, especially if the medical community has a lack of awareness. And because it's so rare, often they don't see too many cases. So that connection of one family member to another is vital in our support efforts, especially in countries where they don't have support organizations. It's even more important. There's criteria to become a member. They have to have a nonprofit company established in their native country. They need to have a board or a committee that's helping guide them as they continue to stand up their organization and expand. They need to align, their objectives need to align with the alliance, and primarily to be committed to offering support to patients, families, caregivers, and those at risk of all forms of prion disease. The other thing that we've done, which has been vital to our success and our support across the globe, is establishing key partnerships. First and foremost, I think I heard Victor reference our friends and advisors. These are experts within the prion disease community. They could be researchers, they can be healthcare professionals that are committed to prion disease, committed to supporting patients and families. And we have, I think, Suzanne, 52-ish friends and advisors that we often tap on the shoulder for assistance. If we get an outreach from a country that doesn't have a support organization, we reach out to our friends and advisors, and they often know of a clinician or an expert in that country that we can then connect the family to. And again, that's critical in those countries that don't have support organizations. So we rely on them often. If a new journal article comes out, we tap them on the shoulder to translate that into layman's terms so that our families can understand what's happening in the research or the new findings that they have. The other key partnership is with surveillance centers where they exist. We encourage our member organizations, especially those that we're starting up and helping with, is if you have a surveillance center, that partnership is vital. You know, I know that Suzanne has great experience with that in her country, as do we in the United States. That partnership, you just can't put a price on that. So that's critical. Healthcare professionals we've talked about. Again, if we don't have a support organization, or even if we do, we encourage them to find those healthcare professionals that are interested or already committed to prion disease, educate them, build those relationships so that if patients or families call and say, we've been all over, we can't find anybody who understands what's happening, we can help direct them to those professionals. Researchers, another key partnership, and most recently pharmaceutical companies, as clinical trials are being discussed, is happening within the next few months. And Suzanne and I have been engaged with them, I'd say for the last 9 months to a year. Oops. Oh, I'm sorry, I got to back up for a minute. So when we talk about our friends and advisors, we want to take a few minutes just to show you a quick video of where we have them in the country, in the various countries, and who some of the names are, and I'm sure you'll recognize some of them. So again, you see all the countries that we've covered so far. We rely on them tremendously, and we're grateful for their collaboration, their assistance to us as an alliance, and to all the support organizations. So where do we go from here? When we formed the alliance back in 2006, and since then it's been informal. And Suzanne and I have talked a lot over the last year about how do we move this forward, especially with the talk of clinical trials. How do we accelerate our work? How do we get more countries established with support organizations for the benefit of the patients and families, but also in the light of potential clinical trials coming up? So there's a few things that we've done this year. First and foremost, we did establish the CJD International Support Alliance as a nonprofit company. Suzanne, myself, and Professor Richard Knight are the company directors. And this does a few things for us. It allows us to really accelerate some of our goals and objectives that we'll talk about, and put some formal structure in place and be prepared to help wherever needed as clinical trials come to fruition. We've also established our board of directors, and we actually had our first meeting at the Prion 2023 conference in Portugal this year. And these names are very familiar to us and maybe to some of you. They are experts in the field of prion disease, and they are very committed and have been friends and advisors of ours for many, many years. So in addition to the three of us, we have Professor Brian Appleby from the United States, Joaquin Castilla from Spain, and Professor Michael Geschwind from the United States as well. So we're very excited about the perspectives that they bring, because they all come from a very different perspective. And that's going to really help us as we expand and accelerate our work. And we couldn't be more pleased to have them on our board. The one key tactic that we're taking now that we're a formal organization is that we can now take in funding, because whatever we've done to this state, Suzanne and I have just done on our own. And what we want to do is create our own support organization expansion and advocacy grant program. And that will really address 4 of our objectives that we talked about, which is to assist countries to establish their own support organization or help those that are existing expand service offerings. It also promotes continued access to care because of the other connections we're going to have in those countries. And then again, in those countries where we stand new organizations up or expand services, that promotes the avenue for patients and families to know about clinical trials, to keep updated on what's happening in research, connections to those medical professionals, et cetera. So what we don't want to do is compete with the support organizations. We want to support them and facilitate their work. So in order to get our grant program up and running, we need funding. But again, we don't want to compete. The support organizations, and again, they vary in where they're at and what services they offer. I think all of them are looking at funding and raising funds for research. So we don't want to compete with that. So we're going to go after company-based grants that the Alliance can get to fund our grant program. And then we're working on our own grant application process and the criteria for our member organizations to apply for the grants so that they can stand up their services. You know, standing up support organizations is not easy. It does take committed, dedicated individuals. It's time, it's resources, including funding. And we don't have a problem with the committed, dedicated individuals because they are very motivated because they're usually impacted in some way by prion disease. It's sometimes it's the time and the money that they need to stand up a nonprofit, and the cost and the rules and the regulations from country to country vary. So we want to help them accelerate their work by standing up this grant program. So we're going to provide seed money to them to get things moving. We are happy to announce that we have been awarded our first grant from Ionis Pharmaceutical. We're very grateful to them that they really saw and believed in our vision, our mission, and our key objectives. And we're willing to support us in this way. We have another grant proposal that's pending, and we plan to apply for other grants in order to, again, support our objectives and help other countries. The other thing that we want to do is to create the Alliance CJD Support Organization Toolkit. Again, we need to get speed to value, and the beauty of being part of an alliance such as this is that we have best practices that have already been established, especially in those support organizations that have been around for a while, like the one in the US, Australia, UK, Spain's doing some really great work in a very short period of time. So we want to leverage those best practices and provide organizations a roadmap and artifacts that they can take and localize to their country. Again, rules and regulations in countries vary, so we can provide a template to them. They can localize it to what their rules and regulations are in their country and to their own language. But we want to not have them start with a blank sheet of paper. We want to give them a jump start, and this is one way that we can do that. This is the roadmap. I'm not going to go over it in detail, but just wanted to show you a little bit about what we're going to provide to them. On the top, across the top, you have the life cycle stages. So someone has the idea they want to do a support organization, or they're in the startup phase, growth, and then they're highly mature. You have organizational elements down the left side, and the last two at the bottom are really how tactics they can use and employ in order to meet their objectives, and what measures and metrics that we're going to want to see if they get a grant from us as to how this is all working and how we're able to impact the prion disease community. So we talk about the impact, the brand, revenue, operation and culture, and governance. And then the tactics are things that we have learned over the years, what kind of you would need to start up and get started as you're growing, things that you may want to add in. And then once you're highly mature, you know, what those pathways are for them. So next steps for us, again, is to stand up our grant program, create the toolkit, launch and award our first grants, which we hope to do early next year. And at the same time, we're working to obtain charity status. Some of the grants that we've looked into want charity status, not just nonprofit status. So those are things that we're working on. We're leveraging our board to really help review what we're putting together. A lot of them have done their own grant writing, so they can help us in that perspective and how to stand up our grant program. So we're excited about the work that we've been able to achieve. We're very excited about formalizing this alliance and really trying to accelerate our work, being able to offer funding to these countries to get things started, because research is critically important and we understand that. But while the research is happening, we still have patients that are dying from prion disease, families that are devastated by never hearing of this disease before, and now they're living it. And that support has to be there. And that's one of our major objectives. And then that research and these clinical trials are just the best hope that I've heard in the 25 years since my mother passed away. So it's a very hopeful time. We're very excited about the potential. We're realistic. We know it's going to take more time. This isn't the end. This is just the beginning. And we're committed to do whatever we can to help them be successful and find a treatment or cure for this disease. Suzanne, do you want to add anything at this point? You're on mute.
No, you're doing a great job, Dina.
Again, this is just the logos of the various members of the Alliance. And we just want to thank you for the opportunity to bring awareness to this group. Awareness really brings about change. And so it's critical for us to continue to advocate and bring awareness. And we appreciate the opportunity to share our story with you today. And we're open to any questions the group might have.
Thank you so much, Dina, for your presentation and for showing us what the CJD International Support Alliance has been doing in order to support the supporters of the families in a lot of the countries. I just realized that Suzanne has been in the CJD support group network for a long time, and they recently got their own meeting and their 30th anniversary. So congratulations for that amazing amount of work that you've been doing. It's not easy. It's very hard work, because this, as you mentioned previously, this comes from the families themselves. And all of these foundations have been established by family members who have in some way or the other been dealing with CJD, with a prion disease, and it is not going with or trying to carry with the burdens of treating a disease such as a prion disease, but again, it's trying to give light to other family members who at the moment can see the road or the way to give a better treatment or opportunities to their own family members. I guess this is a very, very humanistic work that you've been doing. I don't know if the CJD Support Group Network is the oldest or one of the earliest support group networks for prion diseases that was established, but 3 decades is quite a long time, and I believe a good amount of work. Congratulations on that work, Suzanne. Congratulations, Dina. I know that you've also been working in foundations before being at the CJD International Support Alliance. I know that you recently retired from your nursing job, and you are still active helping families, not only in the US, not only your own family, helping families all over the world. And from the Mexican side, and for me as well, I also would like to congratulate you both for all of this amazing job that you've been doing. I don't know, Francisco, if you have some words for them.
Yeah, it's a special thanks. I know that it's a long, long way, very far from here, from Mexico, that you are here with us to sharing with us all the information that you have. And it's amazing. It's a very good work that you have. Thank you very much for sharing us and to Mexico, all the people to Mexico, all this information. Thank you very much.
You're very welcome. We're glad to share, and part of our job is to bring a face to the disease, and you do that by sharing stories and getting out there and talking about it. So we're happy to do it. It means a great deal to us to be able to share and bring awareness, and we're hopeful for good things to come.
Just on
I'm
that,
sure of that.
Just on that, Victor, I believe that the CJD Foundation had their 30th anniversary, founded in '93, as was our support group in Australia and our surveillance unit. And the UK was, I believe, '95. And yes, we did have our 14th annual national conference last weekend, and we did have a celebration honouring 30 years of assisting families. And as some of you know, I'm at risk of CJD from receiving human pituitary hormone. So I'm really proud to say that we are the only organisation that started off with a risk group that has managed to expand and grow and support families. And I think I've worked with almost 1,000 families now, offering them support and help during what is a very devastating and difficult journey.
And this work came to the ears and eyes of the Queen. I was telling the people in Spanish that you received this award a few years ago due to the work and the great effort that you've put assisting all of these families. And as you said, you started in Australia, but now you're all over the world. Congratulations for that effort, and for supporting not only the families, but the friends and advisors. All of the names that you showed in those slides. Well, we do advise, but we also receive a lot of support from you, Suzanne, from you, Dina, and from all of the members in the CJD International Support Alliance. So I guess it is a give and give. And we're very happy and glad of receiving all this support from you as well.
And none of us can do this alone, Victor. It really is a collaborative effort, and to see such a dedicated, it's a small group when you think about other diseases that have a ton of research and other things going on for them. But the dedication of this group of people meeting you and others in the field has been a blessing. And it takes the collaboration amongst all of us and the surveillance centers and families to really get us where we are today, and hopefully propel us into a future where we do have a treatment or a cure.
I hope we do. We thank you again for being here with us in our first symposium of prion diseases in Mexico. You both know that we've been thinking about having these meetings for a long time. We just started. We hope to have in-person meeting next year, and we do hope to have you in Mexico in the next year. We will be working on that. We are showing the diploma in the transmission in recognition of your talk today. We will send it to you by email. And I guess we need to move forward to our next presentation, which is by Francisco, the president of the Mexican Foundation. And you are obviously welcome to listen to his talk. So we will move forward. And I will present el arquitecto Francisco Mota. El arquitecto Francisco Mota ha sido un motor importante para el establecimiento de la fundación en México, que se conoce como la Fundación de Enfermedades Neurodegenerativas México AC. El esfuerzo surge precisamente por apoyar a las personas, a las familias con una enfermedad por priones, pero viendo el enfoque precisamente o el alcance para esta fundación se amplía a abarcar algunas enfermedades neurodegenerativas y no estar limitado exclusivamente a las enfermedades por priones. En esta ocasión, el Congreso, que está enfocado precisamente a enfermedades por priones, tiene este alcance porque recientemente se realizó el CJD International Awareness Day, ¿no? El día para generar conciencia por las enfermedades por priones, que se realizó prácticamente hace unos días, fue el 12 de diciembre, y la intención precisamente, una de las intenciones de esta fundación es precisamente abonar a esta conciencia en nuestro país. Y el arquitecto Mota ha estado trabajando en el establecimiento de la fundación. Es un trabajo impresionante desde el punto de vista administrativo, legal, financiero, y pues estamos ahora aquí precisamente reunidos todos nosotros en buena parte gracias a este apoyo que hemos recibido del arquitecto, y sin más le dejaré yo la voz a él para que escuchemos precisamente cómo ha trabajado, cuáles son las intenciones de la fundación, hacia dónde se quiere llegar. Sí, sin más le dejo la voz al arquitecto Francisco Mota para escucharlo con su presentación sobre la Fundación de Enfermedades Neurodegenerativas México. Muchas gracias, doctor. Arquitecto. Dale, ya te puse el grado de doctor, perdón. Adelante.
Oh, muy bien, gracias. Digo, la verdad quiero agradecer de todos modos también a todas las participantes de todos aquí. O sea, lo voy a decir en inglés. We want to special thanks to all our speakers. Professor Richard Knight, Dr. Michael Heswit, Dr. Eric Minkel, and the special thanks for Gina and also to Susan. Y pues, ¿qué más te puedo decir también? A ti, Víctor, pues muchísimas gracias también. Eres un gran motor también de esta fundación. Tú eres la parte que mueve todo esto. Entonces, te agradezco muchísimo de toda esta participación tuya en esta fundación. Les voy a decir ahorita, de todos modos, una breve introducción básicamente de lo que es la Fundación de Enfermedades Neurodegenerativas México, AC, o también llamada FEDMEX. Es una fundación sin ánimos de lucro, es una entidad cuyo fin no es la persecución de un beneficio económico, principal humanitaria y o comunitaria. Este tipo de instituciones por lo general se financian gracias a ayudas y donaciones derivadas de personas físicas, empresas, instituciones y organizaciones de todo. 32.4 millones de habitantes serán personas mayores por la población 5 se ven afectados por problemas de salud neurodegenerativos. En total, se estima que más de 800,000 mexicanos sufren de alguno de estos padecimientos neurodegenerativos. El humano está evolucionando cambiando sus hábitos y costumbres, por lo que su calidad de vida está extendiendo su longevidad. Pero no todo el cuerpo se adapta. La parte más importante de él, el cerebro, se deteriora y en consecuencia limita al ser humano en sus actividades. Enfermedades como el Alzheimer, Parkinson, Huntington, el CGD, demencia senil, esclerosis lateral amitrófica son las principales en México y en el mundo. Afectan al sistema nervioso y ocasionan pérdidas de memoria, alteraciones de conducta y las funciones cognitivas. Actualmente la Organización Mundial de la Salud estima más de 30 millones de pacientes en el mundo con Alzheimer, duplicándose esta cifra cada 5 años. Durante las últimas décadas se ha producido un cambio hacia la atención de las intervenciones tempranas para maximizar la ventana terapéutica a través de medidas de prevención primarias que actúan sobre los factores de riesgo accionables o medidas de prevención secundarias para frenar la progresión de la enfermedad, afirmaron estos científicos. Especialistas, instituciones para poder ayudar a las personas que padecen estas enfermedades en busca de formas de prevención, tratamiento y, en su caso, la cura. Pero, ¿qué pasa con los familiares de estos pacientes? Son ellos los que cargan con los gastos y cuidados en la mayoría de los casos. Familiares que, aparte de realizar este trabajo, tienen la incertidumbre de portar genéticamente esta bomba de tiempo. FENMEX no se olvida de ellos. Además de proveer apoyo a los pacientes de esas enfermedades, se enfoca en apoyar a los familiares. En la mayoría de los casos, los familiares son los que más sufren y es urgente desarrollar procesos manuales, agendas y canales de ayuda. Para lograr esta sustentabilidad y además estar en la vanguardia de los últimos descubrimientos en este ramo, FENMEX estableció los canales de comunicación con las más grandes fundaciones especializadas en este ramo. Esta unión hace que las investigaciones y resultados fluyan de una forma orgánica, en beneficio de los pacientes. Aunado a este mecanismo de información, también se establecen las fechas de encuentros globales en formas de convenciones o simposios donde la interactividad real y virtual tienen efecto, lo que permite tanto a estudiosos como pacientes compartir sus avances y experiencias. Necesitamos generar la participación de todos los sectores de la población. Los años avanzan para todos sin excepción y es importante no olvidarlo, porque después puede ser tarde para recordarlo. ¿Qué servicios ofrecemos que no existen en otras fundaciones? Podemos dividirlo en 4 grandes temas. Apoyo para los familiares y pacientes, así como el voluntariado que se vaya uniendo. Número 2, la prevención, con la capacitación y entendimiento a familiares, amigos, enfermos, empresas que deseen tener una cultura entre sus colaboradores de estas enfermedades, dar a conocer con mayor detalle las consecuencias de cada una de las enfermedades neurodegenerativas, nos dará una cultura distinta. Y si ya la conoces, te enfrentas a estas sin miedos, a todas ellas. Esto es lo que hacemos básicamente con estos simposios o congresos. El tercero son los congresos nacionales e internacionales. Aquí tenemos la oportunidad de tener a los grandes médicos en cuestión de enfermedades neurodegenerativas con sus avances médicos y o científicos. Y la 4, la investigación en los adelantos científicos y alternativos. Se estarán desarrollando y o gestionando en cualquier parte o rincón del mundo, donde tenemos que averiguarlos. FENMEX, a la fecha, ha atendido a 11 familias de diferentes partes de la República en este primer año de atención. tratando de apoyarlos con la información general y en algunos casos particular. Todos han sido atendidos por el doctor Víctor Sánchez. Se requiere de un equipo mayor de profesionistas para el apoyo, que son un grupo de médicos, psicólogos, psiquiatras, para guiar con mayor facilidad a las familias afectadas. Estamos agregando en breve a mayor personal para ello. Estamos en trámite final ante la autoridad para que nos autorice para poder recibir donativos. Agradezco mucho su participación en este simposio y poder hacerlo cada año y poder dar seguimiento a todos los avances científicos de esta enfermedad. Les agradezco mucho y nos vemos en un año, si todo quiere. Gracias. Víctor.
Muchísimas gracias por estas palabras en las que se refleja el esfuerzo realizado hasta este momento por la fundación. Creo que bueno, diría, vamos muy bien, ¿no? Y sin duda seguimos con la mira en el futuro. Ya estamos terminando este simposio y la meta está en ir preparando el siguiente, ¿no? Hemos visto familias y la meta está en ver todavía más, ¿no? Hemos buscado la manera de apoyarlas, ahora queremos apoyar más a esas familias y todavía a más familias en el país, ¿no? Lo bueno es que estamos siempre con la intención de crecer, de pulirnos también y de ser mejores para nosotros y para brindarles lo mejor a a las personas con enfermedades por Priones en mi país. Muchas gracias, Francisco, por la presentación, por esta ventana que le muestras a la sociedad de qué es la fundación, pero también por la ventana que has permitido abrirles a la sociedad de lo que son las enfermedades por priones, cuáles son los avances en el diagnóstico, en el tratamiento, que vayan conociendo a los investigadores, como se conoce, a las puntas de lanza, ¿no? En esta área de trabajo y esperemos que ese es nuestro esfuerzo, nuestra intención, que el próximo año podamos estar reunidos en persona todos, además de los investigadores, del personal de la salud, las familias, ¿no? Que es uno de los enfoques principales para este tipo de charlas, que puedan hablar con ellos, que puedan convivir, que puedan expresar sus inquietudes y responder varias de sus dudas. ¿Por qué no? Incluso podamos tener ya incluso la manera de participar en alguno de los ensayos clínicos para probar nuevos medicamentos en, en estas familias, ¿no? No sé si gustes dar algunas palabras finales para clausurar este evento, agradeciéndoles a todos los que nos han apoyado, la Universidad de Guadalajara, la Fundación, bueno, todos los nombres que ya nos darás en un momento. Yo desde mi trinchera agradecer a la Universidad de Guadalajara, a todo el equipo de apoyo para tener este tipo de transmisiones, a los alumnos, estudiantes que nos estuvieron también apoyando en múltiples foros para poder tener esto listo y también a la fundación, pero yo diría muy personalmente, además de la fundación, obviamente, a los ponentes, ¿no? Son personas con las que hemos trabajado, al menos yo personalmente, desde hace más de una década, que nos hemos estado apoyando, pero sin duda nos han apoyado más a nosotros que uno a ellos en la formación, en la atención de otras familias, algunos de ellos, como ya lo escucharon ustedes, em, tan pronto les mandé un mensaje de invitación, de inmediatamente en fin de semana, organizando otros congresos, eh, contestaron y estuvieron muy dispuestos para estar con nosotros, independientemente de que para algunos de ellos estas fechas son muy personales o incluso en horarios muy de madrugada, ¿verdad? Estuvieron conectados, estuvieron con nosotros. Yo les mando ese agradecimiento a ellos, de nuevo otra vez a la universidad y a la Fundación Fermex. Y ahora sí, me callo y te dejo la voz, Francisco, para que puedas clausurar este evento.
No, pues la verdad agradecemos a todos los que escucharon, que aprendimos, la verdad, pues varias cosas adicionales que no sabíamos creo que esto nos sirve pues, de mucho, la verdad. Entonces, creo que esto pues, ha sido benéfico para mucha gente, estoy seguro. Para nosotros, obviamente, pues nos enriquece también enormemente como fundación. Y pues digo, ahora sí que cerremos esto, ahora sí que como se debe, agradeciendo a todos, la verdad, todos su colaboración, su apoyo. Hay mucha gente que está atrás de esto. Nombrar a todos creo que es, pues digo, está difícil, la verdad son muchos, pero agradecemos a todos y cada uno de ellos. Y a todos los que escuchan este simposio también, espero que les haya servido como a nosotros nos ha servido. Pues muchas gracias y adelante. Nos vemos el año que entra.